지금 저 모델로는요, 지금 동영상 촬영 시에 측광 모드 변경이 안 됩니다. 사진 촬영 시에만 변경이 가능하고요. 그래서 이쪽에 강제 노출 보령 넣어줬어요. EV 가로수로 플러스 2로 했어요.
네, 항상 보시면 네. 어쩔 수 없어요. 상황에 맞춰서 갑시다.
예, 지금 한번
좋아요.
이럴 때 쓰는 거지. EV 다이얼이.
Good morning, ladies and gentlemen. Welcome to the AIxBIO Global Forum. Please complete your registration at the front desk if you have not completed, and find your seats. We'll be starting promptly at 9:30. 아니요? 전 사회잖아요. Ladies and gentlemen, good morning and welcome to the AIxBIO Global Forum. My name is Sungwon Jung and I'm the chair of the organizing committee of Korean Society for Bioinformatics. It is my great pleasure to be your host today. To begin, I would like to invite Professor Sungwoo Ryu, President of the Korean Society for Bioinformatics, to give his opening remarks. Please welcome Professor Ryu.
Good morning. Ladies and gentlemen, distinguished guests, and esteemed colleagues, on behalf of the Society for Bioinformatics, it is my great honor and pleasure to welcome all of you to the AIxBIO International Forum. And special thanks to co-organizers, Ministry of Science and ICT, National Research Foundation of Korea, Korea Institute of Science and Technology Information, Korea Bioinformatics Center, and Korea Bio Research Association, and Hanyang Institute of Bioscience and Biotechnology. We stand today at the forefront of the remarkable era where artificial intelligence and bioinformatics are converging to redefine the possibility of science, medicine, and human health. The integration of AI with biological data is driving a paradigm shift that is transforming how we understand life, diagnose disease, and design therapeutic solutions. Over the past decade, bioinformatics has evolved from a supportive discipline into a central component of modern biomedical research. And with the emergence of AI, its potential has expanded exponentially. This forum serves as a global platform for leading experts, researchers, industry leaders, and policymakers to come together and share cutting-edge insight, best practice, and vision for the future. I would like to express my sincere appreciation to our organizing committee, distinguished speakers, and all the participants who made this event possible. As a chair of Society for the Bioinformatics, I'm proud that our community continues to lead these transformative conversations and contribute to shaping the global agenda on AI in life sciences. Let us take this opportunity to learn from one another, challenges assumptions, and build lasting partnerships that will drive scientific progress. Thank you once again for joining us, and I wish you all a successful, productive, and inspiring forum. Thank you.
Thank you, Professor Liu. Before the welcome speech, let me briefly introduce our distinguished guests. Mr. Seung-rae Lee, Executive Vice President of the National Research Foundation of Korea. Dr. Sik Lee, President of Korea Institute of Science and Technology Information. Mr. Ji-soo Son, Assistant Director of the Ministry of Science and ICT. Oh, he's running late today. And Professor Sun Kim from Seoul National University. Professor Predrag Radivojak, President of International Society for Computational Biology. Professor Bonnie Bogle has arrived here. Oh, she's coming late. Okay. And also Ms. Diane Kovas, CEO of AIxBIO, but she's also running late, I guess. Now, we will have welcome speech of our guests. Firstly, Mr. Pan-sik Hwang, Deputy Minister of the Ministry of Science and ICT, was unfortunately unable to join us today due to unforeseen circumstances. In his stead, Professor Jinwoo Nam will deliver the welcome speech. Please welcome Professor Nam.
Good morning, everyone. My name is Jinwoo Nam from National Research Foundation of Korea. I'm a director of Advanced Biotechnology Division. Unfortunately, as already told, Deputy Minister of R&D Policy Hwang Pan-sik from the Ministry of Science and ICT could not attend this forum because of the urgent schedule at the National Assembly. So on his behalf, I've been asked to lead this congratulatory remark, and I'm really honored to do so. Okay. Okay. So you can read English and Korean together. So distinguished guests and esteemed experts who are shaping the future of the AI bio industry, it is great pleasure to welcome you all. I'm Hwang Pan-sik, Deputy Minister of R&D Policy at the Ministry of Science and ICT of the Republic of Korea. First of all, I'd like to sincerely congratulate you on the hosting of the AIxBIO Global Forum 2025. I extend my heartfelt gratitude to Professor President Ryu Sung-ho of Korea Society for Bioinformatics and to all the organizers who made today's event possible. I'd also like to thank each and every one of you for joining us today despite your busy schedule. Today's forum is a meaningful opportunity to explore the future of the convergence between AI and biotechnology. And discuss how we can shape the future of AI bio ecosystem while seeking out its direction. AI is no longer just a data analysis tool. It has become a transformative partner in life science research. In areas such as protein structure prediction, genome analysis, and virtual screening, AI is significantly reducing research time, cost, and solving problems once thought unsolvable. The potential of AI is in drug discovery, precision medicine, and digital healthcare goes for beyond imagination. And already global competition is accelerating rapidly. To respond to this trend, the Korean government is developing the AIxBIO expansion strategy with full-scale effort. So this strategy spans the entire innovation cycle from R&D investment, data infrastructure, and talent development. Korea envisions becoming a global AIxBIO hub. To support this vision, we are already actively implementing a range of important initiatives. Through the K-Melody project, we are building a federated learning-based platform to support AI-driven drug discovery. The National Integrated Bio Big Data project is collecting and utilizing large-scale biodata, including genomic and health information, to accelerate precision medicine and disease research. We are also operating KBDS, the Korea Biodata Station, a national platform that integrates and connects public and private sector bioresearch data. Furthermore, to establish a strong regulatory foundation for innovation, we have enacted the world's first Synthetic Biology Promotion Act, which will position Korea as a global leader in the emerging field of synthetic biology. AIxBIO is not merely a fusion of two technologies. It is a bold step toward defining the future of health and life itself. I hope today's forum will be a source of wisdom and inspiration that bring this future close to us all. This Ministry of Science and ICT will work closely with the private sector, academia, and research community. We will also expand global collaboration so that Korea can rise as a true leader in AIxBIO. So once again, congratulations on the successful opening of the AIxBIO Global Forum. I wish you a productive and engaging discussion. Thank you.
Thank you, Professor Nam. Now, I would like to invite Mr. Seung-rae Lee, Executive Vice President of the National Research Foundation of Korea, for his welcome remarks.
네, 반갑습니다. 제가 영어로 준비했지만은 혹시 주변에 옆에 외국인 계시면 교류의 장이니까 해석을 좀 해주시기 바랍니다. 네, 가능하시죠? 그러면 저는 우리말로 하겠습니다. 자, 안녕하십니까? AIxBIO 글로벌 포럼에 여러분을 환영하게 돼 정말 기쁩니다. 저는 한국연구재단의 사무총장 이성래입니다. NRF 그러니까 한국연구재단은 기초과학과 공학부터 인문사회과학까지 전 분야의 학술 및 연구개발 활동을 지원하는 대한민국 대표 연구 지원 기관입니다. 2009년부터 우리 세 개의 주요 재단이 통합되어 설립된 NRF는 혁신을 촉진하고 인재를 양성하며 국제협력을 증진하는 것을 목표로 하고 있습니다. NRF 내에는 국가전략기술본부가 있으며 이는 우리나라 미래의 중요한 기술을 발전시키는 데 핵심적인 역할을 합니다. 그 산하에 세계의 바이오 기술단은 생명공학, 의학 분야의 첨단 연구를 지원하며 국민 건강을 증진하고 경제 성장을 이끄는 것을 목표로 합니다. 오늘 이 자리는 전 세계에 모인 인공지능과 생명과학 분야의 최고의 전문가들이 함께하는 뜻깊은 자리입니다. 이번 행사를 기획해 주신 한국생명정보학회에 깊이 감사를 드립니다. AI와 바이오가 어떻게 과학과 건강 분야의 큰 문제를 해결할 수 있을지에 대한 다양한 이야기가 정말 기대됩니다. 이 포럼은 단순히 지식을 나누는 자리가 아니라 연결을 만들고 협업을 촉진하며 미래를 함께 상상하는 자리라 생각합니다. 여러분이 과학자든 정책 입안자든 학생이든 오늘 포럼이 새로운 생각과 협력을 이끌어내는 계기가 되길 바랍니다. 다시 한번 참석해 주셔서 감사합니다. 멋진 포럼을 만들어 봅시다. 네, 감사합니다.
Thank you. Now, please welcome Dr. Sik Lee, President of Korea Institute of Science and Technology Information, for his welcoming speech.
Hello, my name is Signi. I'm the president of KIST. So there was some miscommunication between our staff member and this society. So my secretary prepared the greeting message in Korean. I'm sorry. So I just translated it to the English with the ChatGPT. I found 90% is acceptable. So I say both Korean and English together. Okay. Okay, but too small. Distinguished guests, ladies and gentlemen, it is a great honor to welcome all of you here today. I would especially like to acknowledge the presence of Dr. Ryu Sung-ho, President of the Korean Society for Bioinformatics, Dr. Frederick Ladibojek, President of AIxBIO, and Dr. Bonnie Berger, Chair of the RICOM Steering Committee, Dr. Sung Lee, Secretary General of National Research Foundation of Korea, as well as the scholar, researcher, and industry expert from around the world in the field of bioinformatics and artificial intelligence. Welcome. Personally, I was a founding member of Korean Society for Bioinformatics, and I'm feeling comfortable as if I'm coming home. First, I sincerely congratulate you on the successful hosting of the AIxBIO Global Forum 2025. I extend my deep appreciation to the Korean Society for Bioinformatics and all related organizations for arranging this meaningful event, and to each of you for taking time of your busy schedule to attend. Today, we are living in an era where the remarkable convergence of artificial intelligence and biotechnology is creating a pivotal turning point in improving human health and quality of life. AI-powered genomics, precision medicine, drug discovery, and protein structure prediction are advancing at speed, and accuracy levels once unimaginable, thanks to the combination of rich life science data accumulated over decades and cutting-edge AI technologies. At the Korean Institute of Science and Technology Information, we are national center, so we are in charge of national supercomputing infrastructure, research network, and data accumulation and data management. And recently, we announced that is AI and quantum computing and bioinformatics our key, our focus areas. So we are trying to more invest on this area. This forum goes beyond academic exchange. It serves as a significant platform for experts from diverse fields to come together and discuss global scale collaboration and strategy. I hope this occasion will become a key turning point for the future of bio research, where the boundless potential of AI and bio convergence is explored, and where research institutes, industry, and policy bodies, both domestic and international, seek direction for innovation together. Finally, I wish this forum to be a truly inspiring and insightful experience for all participants. And I sincerely wish you continued success and growth in the AI bioconvergence field. Thank you.
Thank you very much, Dr. Lee. Now, we would like to invite our distinguished guests, session speakers, and chairs for a group photo on stage. So please join at the front. Yes, please. It's all done, or? Oh, it's a very long photo shoot. Okay, thank you. Actually, we are going a little bit early on schedule. Anyways, we'll now take a coffee break. Please enjoy the refreshments behind you, and we'll resume the session promptly at 10:15. Thank you. And if we have interest in taking further group photos on the floor, so anybody can welcome here. So please feel free to come on the stage, and we'll provide a chance to make a photo shoot. Feel free to come on the stage, and we'll make a photo shoot. We'll also make a final photo shoot at the end of our program, today's program at the end of today. But we'll have one more chance here now, right now, for everybody's photo shoot.
네, 감사합니다. 예. 이게 포인터가 안 되죠? 안 되니까 잠깐만 이거 제가 화면을 이쪽을 볼 수 없을까요?
아, 이쪽으로 보시면
이게 포인터가 있으면 여기 포인터라도 쓸 수 있으면 좋은데.
잠시만요.
예. 예, 안녕하세요. 네, 안녕하세요. 네, 안녕하세요. 성함이 네네. 예. 경제학으로 이렇게 좀 바쁘신데.
아, 이 자리가 없어가지고
예.
내일 있어가지고 추가시키려고 나왔어요. 내용이 좋아서.
네. 이게 포인터가 횟수가 있으면 좋은데 안 되면 뭐 그나마 이게 있어야 돼요. 예, 마우스.
지금은 화면이 잠시만요. 제 마우스 화면이 마우스로는 확인하실 수 있으시죠?
예. 마우스라도 쓸 수 있으면 좋습니다. 됐습니다. 예. 이거라도 있어요.
아, 예, 알겠습니다.
everyone. Now we are going to start our session. So welcome back. Now it is my pleasure to introduce Professor Sang-woo Kim from Yonsei University, who will chair our plenary talk.
Thank you. I'm Sang-woo Kim from Yonsei University, and it's my honor to chair the first plenary talk that opens this forum, which will be made by Professor Sun Kim. So I'll introduce Professor Kim. Professor Kim majored computer science, and he got his PhD in University of Iowa in 1997, and he worked as an assistant and associate professor in the School of Informatics and Computing, Indiana University from 2001 to 2011, and he moved to Korea, and he joined Seoul National University in 2014, and he has been a professor in Seoul National University in the Computer Science and Engineering Department. So please welcome Professor Sun Kim.
Thank you very much for the kind introduction, and this is an honor to share my opinion and my experience at this forum. And I'm going to talk about how can AI bioinformatics can do for you to advance biology, medicine, drug discovery. Okay, so scope of my talk is, fortunately my talk is not very technical. I looked at the title of talk after my talk, and then they are technical. So my talk is not technical. So, however, instead I'm going to talk about strategies on how to utilize state-of-the-art AI tools and database, because I have to cover. I asked to cover many topics for you, and then, so main topic of this talk is how we model relationship. So please remember this keyword. The relationship is keyword for this talk. The agenda is I'm going to start with Deep Learning 101 because how deep learning deals with the relationship. And then there are so many pre-trained models. So with the pre-trained models, navigating chemical and molecular cell disease research on multimodal space, the reason I say navigating is that deep learning makes a vectorized space, which we can navigate in the space. So Deep Learning 101, this is a slide from my student at the AI conference. And then look at the two sentences. Eric books flight. And he gets ticket and read books. And then two syntactic, the two same word, books. However, the semantics of this word is different. When you mapping this word into the space, we need to maximally separate these two books instance. And then also, when you look in the flight and ticket, yeah, they are related. So in the space, they have to be in the as near as possible in the embedding space. That is the point of the the making the space. And then important thing is that we need the context need to be modeled in vectorized representation of the word. And then even though we use context to model relationships, sometimes it's pretty difficult. For example, look at the two sentences. The animal didn't cross the street because it was too wide. The other sentence, the animal didn't cross the street because it was too tired. If you pay attention, only one word is different. Wide, tired. When you ask them, what is it? Without focusing on the wide and tired, we cannot answer these questions. So this is attention. This attention is basic technology for transformer, and then this is basic technology for ChatGPT you guys are using right now, actually. So with these pre-trained models, it's very useful. You enjoy using ChatGPT and Gemini, and then going more than the data, we can use a chain of thought. We make a kind of, the inference on that, meta-learning, transforming, and those many things. Now we want to use this one for deep learning technology for medicine and AI technologies. Like a large language model, modeling relations is the key task, basically. Okay, that's what I'm trying to explain today. So, however, there are challenges. What is the meaning of the cat? Are you kidding? Cat. What is this image? Are you kidding? Cat. What is this chemical compound? I don't know. Even medicinal chemist, if she doesn't deal with this chemical compound, doesn't know what it is. This is gene expression profiles of cancer patient. What is it? Even cancer doctors cannot interpret this one, right? Point is that the subject we are dealing with, chemical compound transcriptome profiles are not easily interpreted. For example, look at the English word cat, image cat. We know, everybody know. However, chemical compound expression profiles, nobody know actually. That is our challenge to vectorize this information, okay? So that is the goal. So strategy is that whenever you cannot interpret something, you need to do multi-view. As many perspective, look at the object and then vectorize, okay? So drug discovery and personalized medicine. Drug targets protein, and because as a result of protein, complex gene interaction network will be perturbated. And then patient as a collection of cells, and patient phenotype will change as a result of drug treatment. This one going to deep learning, we need to vectorize everything. Vectorize chemical compound, vectorize proteins, vectorize cellular network, vectorize patient. Can you do that? This is the topic of my talk. Yes, we can. Not me, actually. Around the world, many researchers work very hard. Now it is possible. So let's begin. So here's my talk agenda. I'm going to start with there are two important topics. One is the knowledge graph. The other one is the pre-trained models. And then important topic for this one, when you apply this one, is that shortcut learning, which means that trained model doesn't work on your data. That's common. How can you overcome this, challenges that we have to use transfer data, transfer learning with your own data. How you embed your own data in such a space, and then how we augment data is a keyword. Also, you guys using large language model, and large language model, two different points. One is a large language model, like a transformer, is a technology, simply, or LLM, like for example, GPT, it's just a computational framework. I'm going to show you how it does, actually. So chemical space navigation, the reason why I use word navigation is, again, deep learning vectorize everything. As a result of vectorization, we have a space. Then we begin to navigate, okay? So pre-training chemical compound, fortunately, it becomes saturated. So the important models, what we are using is Morphomer, Nature Machine Learning Intelligence 2022, Graph MVP ICLI 2022. After this, it's begin to saturate, which doesn't mean that molecular vectorized representation becomes mature. We just need to utilize it. So this pre-trained model representation is very useful. However, this is another representation of your data. And then use of a compound representation for your prediction task requires your own model. Okay, it's simply new types of data for your prediction, actually. So I told you that my talk is not technical, but because I need to show you examples, so I'm going to use two task examples from my experience. One is drug target affinity prediction, and the other one is prediction of drug likeness from ADMET perspective. This is our ISMB paper this year, and then mixing DTA improved the drug target affinity prediction by extending mix-up with the Git by association. To do that, what we are doing is that, yeah, we have a chemical compound, we have a protein. We don't use this one as is, so we use a pre-trained model. One is a Molformer, and then ESM3. And then we have a vectorized representative in the context of big data, because we are using pre-trained data. And then using this one, we have efficient attention mechanism, and then we predict. However, we don't have enough data. So what it does is that we do the C mix-up, so that we mix up by similar proteins, we mix up by similar drugs, and something like that. So we basically increase amount of our data by mixing up. And then it shows that drug target affinity prediction can be efficiently done, actually. The other ISM paper we had this year is that ADME drug likeness, enriching molecular foundation model via pharmacokinetics guided multitask learning for drug likeness prediction. Wow, wording is difficult. And what it does is that we use molecular foundation model, graph MVP, Morse-Error, and graph attention transformers, so that chemical compound becomes vectorized. This vectorized representation will be recalibrated using the absorption, distribution, metabolism, excretion, assay information. So assay data will be used to recalibrate this representation, so we have a new representation. So now we have molecular vectorized representation from big data and then also assay data together, and then using this foundation model getting here, and then we begin to distinguish chemical compound is a drug or not drug. And then we kind of achieved quite good performance. So we had the ISMB paper this year also as well. So this is the part of a technical talk of my, and going to molecular biology space, amazing. So many colleagues around the world are doing a lot of jobs. For example, the DNA. EVO2 has a really good, the pre-trained models for DNA. I think Peja will talk about, the mutations today. RNA transcriptomic, and then there is also a lot of, gene expression profile pre-trained models. Protein, of course, AlphaFold. And then epigenetics, yes, we have pre-trained models. And then cell, we have pre-trained models. I'm going to show you. EVO2, I think many of you know that this is a pretty exciting recent development of the mutations. And then this is our work on Nature Communication. Histone code also can be vectorized by doing the transformer technology. DNA methylation also can be vectorized. And this is a gene former trained on when they published it about 2 years ago, they trained on 30 million single-cell transcriptome data, they vectorized. And then now I think they are using over 90 million single-cell transcriptome data. So when you have gene expression profiles, you can just bring in the pre-trained model, and then it will be mixing of your small data, and then over 90 million expression profiles will be mixed together, simply using these pre-trained models. This is also our example of the ISMB paper last year, pre-training links data for gene perturbation. This is a pretty difficult task because links is different dosage, different time point. We measure the transcriptome profiles without phenotype information. And then using pre-training this links information, we showed that drug response prediction can be very efficiently done, actually. Okay, so that's pretty nice. And then we now, using this one, pre-trained model for gene former and pre-trained model for the gene-gene interaction perturbation as a two pre-trained models put together, and then we are translating cell line level drug response to real patient level drug response, and then we are writing paper about that. Okay, so this is how we use pre-trained models. Proteins, No question about that. Protein as a sequence, there are many pre-trained models. Protein bot, Unimod bot, protein GPT, structure-based AlphaFold 1 to 3, ESM fold 1 to 3. And then also antibody, antibot, AIxBot. We have so many pre-trained models. Simply, our task is how we utilize this valuable pre-trained model because they are trained on the big data, basically. And then your small data will be augmented with the big data if you use pre-trained model correctly. So one of the exciting examples is that this is a Stanford researchers published last year. Directed evolution. When people design the protein sequence, we do the random mutation and then do the biological experiment. And then whenever mutation results in the beta activities, we pick them up and then we go next round. Problem is that random mutation does not convincing. What they did is that they use pre-trained protein models, and then it shows that if we input the current their sequence of interest, they show that which amino acid should be modified, mutated with different probabilities. Using this information, they showed that with a shorter round and better antibody can be generated easily. This is in last year Nature Biotechnology paper by Peter Kim at Stanford University. Now, I talked about DNA, RNA, epigenetic proteins, and then putting together, yeah, we have a cell. We are getting there, actually. Of course, a lot of people know that single-cell bot, and then single-cell bot is latent gene-gene interaction information is modeled as vectorized. And there's a large project called the Human Cell Analysis going on right now. Exciting thing is that this is this year, January, editorial in Cell, how to build virtual cell with artificial intelligence. The modeling the virtual cell has been around more than 30, 40 years. Not successful. Why is that? Because we didn't have enough data. We didn't have enough computational technology. Now, we have data. We have a powerful computational technology. We begin to talk about really we can model cell. You believe or not, we are getting there right now. How? Left panel is a physical cell. Right panel is an AI virtual cell. As you can see, everything can be vectorized. Whenever it's vectorized, creating Cartesian product of vectorized space is much easier than just dealing with the mutation, transcriptome, epigenetic modification, something like that. So we can build virtual cell. We are really getting there right now. If you don't believe me, and then this is another Nature paper this year, foundation model of transcriptome across the human cell types. They pre-trained 2013 human, fetal, and adult cell types. What happens is, look at the title. This pre-trained model achieves experimental level accuracy in predicting gene expression even in previously unseen cell types. Wow. I didn't see the cell. Just knowing the DNA, RNA, protein, those ones, now we are kind of modeling the cell without looking at the cell, actually. So now we are getting there. It is very exciting. This one is actually reproducing experimental, biological assay experimental data without doing experiment, actually, basically. That's the message for this one. Now we are getting there more. Yeah, we modeling cell. Next round is what? I didn't highlight this. Cell-cell communication. I think Professor Jaekyung-won is here, and he actually has a very nice result about this one. Mapping cell-cell interaction from spatially resolved transcriptome data is possible right now. It's vectorized, okay. Everything is vectorized right now. When you're going to disease space, it's complicated, right? Because DNA, RNA, protein, those ones are real entities. Disease is not real entities. I'm just sick, actually, basically. Okay, so this one, the embedding vectorization, should be created only in relation with the scientific article, EHR, phenotype, multi-omics drugs, all those ones. So it's a very complicated story. Then we need help. Help is one is knowledge graph. The other one is large language model can help you. Okay. Yeah, we have scientific articles. There are so many articles, so especially because in Korea, Professor Seoul Gangnam at Korea University created BioBot, which is the most representative vectorized representation of scientific articles. And I'm going to show you a really interesting example. This is not published result. It's just an archive paper, but look at this title. Sensitive Cancer GPT, leveraging generative large language model on structured omics data to optimize drug sensitivity prediction. What? Just to use language model GPT, actually they use GPT-3. Don't do experiment. And then just simply doing the Q&A, prompt guiding, they can predict the drug response. Wow, that is actually a trend. So large language model like GPT, actually new computational platform right now, actually. So how they does that? Drug sensitivity omics data. Botagenetic is cell line, and then they have a mutation, those genes, and then sensitive. And then they translate this into English. And then we did some few shot, not we, they did some few shot training. And then they can actually show that their drug response prediction, don't do anything. Just a simple Q&A prompt engineering, they can predict drug response. So large language model itself becomes very valuable computational framework right now. Okay. Now, going to next topic, knowledge graph. Because putting together graph gene pathway disease is very complicated. Then a lot of people work very hard, especially Stanford at Leskovec, and then also Harvard at the Marinka Jitnik. They did a really good contribution. And then they have a graph, knowledge graph, putting together. And then this putting together information can be also very valuable. Let me show you. First of all, this is our work. It's a biomedical knowledge graph running for drug repositioning by extending guilt by association to multiple layers. Title is difficult, but it's simple. What it does is, given knowledge graph, we do the random walk. We generate many random sequences containing drug and disease together. However, because there are too many genes, putting drug and disease together is not frequently happen. So what it does is using guilt by association, similar drugs, similar diseases put together, we jump and then generate many sentences. Simply, once we generate a sentence, we can easily vectorize. And then that vectorized information, we just put them, drug and disease, in the single vectorized space, and then measure the distance between drugs and disease. Similar drug, repurposing drugs should be closer in embedding space. It simply does that, and we show that this can be done, actually. And Marinka Jitnik, and then she is great, actually. So she was in the knowledge graph, and then now recently is an archive paper, and then she's probably the first one putting together knowledge graph and large language model put together. I showed the large language model can be platform, and then she showed that this can be put together. And then this is amazing, I think, potential right now. And also now going forward, and then there's another article called the TXGNN, which is that modeling heterogeneous knowledge graph using graph neural network technology. This is truly difficult because knowledge graph is a very heterogeneous graph because we have drugs, genes, pathways, and disease. Their entities are pretty difficult, and they show that they can be modeled using graph neural networks. Now, multimodal deep learning technology deployed at healthcare systems. Wow, entire hospital-wise, and then they can be put together. EHR, medical imaging, genetics, voice, everything put together. I'm not saying that it happens, But if you don't believe me, very recently, FDA announced plan to phase out animal testing requirement for monoclonal antibodies. How? They recommend the use of advanced computer simulations, AI technologies. So now, FDA is a very conservative organization. Now they are getting there right now, okay? So what happens is probably a good example is the NIU hospital. Health system scale language models are all-purpose prediction engines. They actually deployed large language model at the NIU hospital. They showed that it's useful. So its scale is really big right now. And putting go together, this is just an opinion vision paper, unlocking precision medicine, clinical application of integrating health record, genetics, and immunology. So disease-wise is happening, healthcare system-wise is happening, and they have recently there is opinion papers making shiny object illuminating. The promise and challenge for large language models in the United Health systems. So now this vectorized representation going to health system-wide. It's not individual DNA, protein, something like that. So they are talking about these titles, actually, basically. So to make my talk short, and I'm going to conclude my talk with a summary. Significance of pre-trained models data. They are new types of data, basically. Okay, we have to utilize. And then in the vectorized space. And then also, LLM becomes a new computer, new computational models, basically. And we have so many pre-trained models, and we can use LLM platforms. We are rich. We have so many. How we can utilize this valuable resource for your research, for your task, is your job, actually. So let's work on this one hard, basically. Summary 2 is how we utilize pre-trained model. You look for the pre-trained models for your research. And then, of course, there's something missing. And then you pre-train your own model for missing ones. And then fine-tune with your assay data. I think this is very important. With your assay data, you have to fine-tune. And then it will be pretty interesting. And the reason why I'm talking about this, then multimodal data integration. For example, many of you tried to have multi-omics data integration, which is very difficult. However, when you vectorize everything, because it's a vectorized representation, integration will be much easier, no doubt about that. So that's why they are talking about virtual cell right now, actually. So finally, perspective is that, yeah, we have a drug discovery paradigm like this one. Yeah, we can vectorize everything because around the world, so many people working on this technology. Indeed, as I showed you, almost everything can be vectorized. Now we have vectorized data. We put together a joint distribution. And then we'll solve your problems in the future. Okay, so I think that's the end of my talk. Thank you very much.
Thank you, Professor Kim. We don't take questions. Okay, it's a plenary talk. So thank you. So I'm closing this plenary session, and we are going to have a keynote speech.
Thank you, Professor Kim. Now, let me introduce Professor Jinwoo Nam, Director of National Research Foundation of Korea, who will chair the first keynote talk.
Good morning. My name is Jinwoo Nam. Actually, I'm working in the National Research Foundation of Korea. I'm also a professor in Department of Life Science at Hanyang University. Welcome. We didn't have a photo with Bonnie. Bonnie just arrived here. Yeah. So it is my great pleasure to introduce today's distinguished speaker, Professor Predrag Radivojac. Professor Radivojac is a renowned expert in computational biology and machine learning. He currently serves as professor and associate dean of research at Northeastern University, Coley College on computer sciences. Professor Radivojac research focuses on the understanding life at the molecular level, particularly how molecular events influence higher level phenotypes. His work encompasses protein function prediction and genome interpretation, mass spectrometry, proteomics, and precision health. In the area of machine learning, he addressed challenges in semi-supervised learning, structured output learning, and domain adaptation. His contributions have been recognized with numerous accolades, including the National Research Foundation of Korea Award in 2007. He is a fellow of International Society for Computational Biology and currently serves as its president for the 2024 to 2027 term. He has also held prestigious positions such as August Wilhelm Skier Visiting Professor and Honorary Fellow at the Technical University of Munich. Please welcome Professor Predrag Radivojac.
Thank you very much. It's great to be here in Seoul. It's great to be in Korea, and it's been a very, very nice time so far for me visiting Hanyang University, and tomorrow I will be visiting Seoul National University. My goal today is to give you a very simple story about how computational tools are currently used and how we are modifying them to be even better used in the space of rare disease. So here's the framework that we have. It's about genetic diagnosis in rare inherited disease. So this is a global problem. There's about 8% of the population, depends on how you define rare diseases. About 8% of population is diagnosed with rare disease. And today when a patient comes to the clinic, the doctors will come and look at the patient, maybe provide some kind of diagnosis for that patient. And then the patient undergoes genetic testing, and there needs to be a genetic diagnosis of that patient. So that is the first key step of genomic medicine, of the precision medicine, that we find the variants or mutations in this patient genome such that we can then proceed with targeted therapy that depends on that variant. But now, even with full genome sequencing, we can only diagnose right now 50% of the sequenced patients. So that is still a very low rate of diagnosis. And the goal, again, is to find one or a couple of variants in this patient genome that are causal in this space. That's a long process. It takes 5 years on average, and it takes a lot of effort by many, many different people. It takes a huge toll on patients as well as on their families. I just want to bring a couple of important things about genetic diagnosis. And so the first one is absolutely it alters clinical care. And so if a patient has a particular variant, there might be therapy designed for that variant, but that is still a minority of cases. In most cases, it provides resolution to the family. For example, a patient would come, or a mother would come and say, what did I do wrong here? What happened to my child? Did I do anything wrong in pregnancy, etc.? But the person may not have done anything wrong. It just happened by chance. So resolution is very, very important. Family planning for future kids is very important, and of course surveillance for other members in the family, for example, more frequent mammography scanning and other types of procedures. And actually, if you're interested in that aspect of genetic diagnosis, there's a very interesting Netflix series called Diagnosis. I'm not here to recommend it, but I watched a couple of episodes, and it really gives you a good sense of what happens. Now, why do I talk about genetic diagnosis? Is because these variants are returned to the patients as official diagnosis. So this is not simply finding predictions. It is not modeling life. It's something that comes back to the patient, and there's quite a different responsibility in that space. So everything that Sun was talking about in previous talk comes to the next level when it hits the patient. You have to actually deliver something to the patient. So what variants can we give to the patients back and say, this is your diagnosis, and be sure that we are not wrong? And who decides on that? So that is going to be my story, and what is the role of algorithms in all of that? So the story comes with a paper published in 2015 by American College of Medical Genetics and Association for Molecular Pathology, so-called ACMG and AMP organizations that have come up with guidelines on what makes a variant pathogenic and what makes a variant eligible for diagnosis. So in that paper, they provide a couple of very important things. So the first one is say, okay, we're going to provide an official definition, what we think is a pathogenic variant. So that is a variant that will be eligible for diagnosis. So here they define something called likely pathogenic variant, and they say if there is a 90% certainty that it causes disease. And then there is also 99% certainty if variant is pathogenic, and the similar holds for benign variants. They also say in this paper that variants that are declared pathogenic by this body, that those variants are clinically actionable. Okay, so that is this committee, this group of people has decided that's what we think is safe to be used in diagnosis, and the practice later has shown that this in fact is safe to be used. So I want to show you how that system works and what are the flaws of that system. There's many flaws of that system. So we are still a long way from providing proper medical care, but it's better than nothing at this point. So this is how it works. So this is an evidential framework, and for every variant, there's a collection of the type of evidence, strength of evidence, and direction of evidence. And then those three things are combined to make sure that the variant can be called pathogenic or benign. So what could be the type of evidence? For example, you might run a functional assay on a variant, let's say a mouse model or a zebrafish model if you're studying developmental disorders and so on. You can have a pedigree and look what happens within a family and so on. Then there's a strength of evidence, which they declare to be in particular gradation here, starting with indeterminate, which means we don't know, there's no evidence whatsoever, supporting, moderate, strong, very strong, and standalone. Now, standalone will not be used in rare diseases. This is only used for common variants to exclude them from potentially diagnosis. And there's direction of evidence that can be pathogenic or benign. Again, benign variants will be the ones excluded. So if a patient has a couple of variants that are benign, they will almost certainly not be reported. So those are used for exclusion, and those that are pathogenic will be candidates to be reported to the patient. Now, for every line of evidence, let me see if I can get my mouse. Okay, it's not working, but on the right-hand side, you can see that a combination of a couple of different types of evidence, at least two, can be used to call a variant pathogenic all the way to the right, likely pathogenic, and then from the bottom, benign, likely benign, and everything in between will be called a variant of uncertain significance. So if you look at the top line, there's one thing that says PS, that is supported, that is pathogenic evidence level strong, and the PVS means pathogenic very strong. So when you combine two lines of evidence, one strong and one very strong, that is a pathogenic variant. So it's a simple rule-based system that you combine different types of evidence and you can call a variant certain type. So if you go, for example, to the last line of the pathogenic, you will see 4 PP types, which is pathogenic supporting. Those are 4 green rectangles. Then one is moderate and one is strong. So combination of these lines of evidence will also give you a pathogenic variant. So that is the framework. That's how it works. And now the question will be, what are the computers here? What is the role of algorithms here? How do we use them? As you can see, in the types of evidence, we have computational evidence. So actually, even right now, when we do genetic diagnosis, the algorithms play a role. So I want to describe you what kind of role they play, what's wrong about that, how can we improve it, and what our role was in this. So this is from the paper, again, from Richard's paper, ACMG AMP guidelines. And you can see they define what can be very strong evidence for pathogenicity, strong evidence, moderate, and supporting. And one that I want to point out is, let me see. It is not coming. I think my computer got stuck. thank you. So sorry about that. So this is actually the next slide. So I'll just explain on this one. So this is for benign evidence. So you can have standalone, strong, and supporting evidence for benign. And what is here circled in the red rectangle is the fourth rule for benignity, which is called BP4. The rule for pathogenicity that I will talk about is called PP3. So this says that multiple lines of computational evidence suggest no impact on gene or gene product. Basically, you have to run two different algorithms or more, and if they agree, then you can, that this is a benign variant, then you can say that this is supporting level of evidence. Now, the committee at the time, in 2015, when this paper was published, did not have a huge belief in algorithms. What they decided is they will give it the lowest level of strength, supporting. And one of the questions that we had is this correct? How did they come up with that? And they came up with that is basically we talked to them and they said, well, in the late 2000s, the algorithms were so hyped that everybody thought that this is not correct. And clinicians actually did not have any faith in it. And by the time they were making these guidelines, they said, we don't have much faith in it. We'll just give it the lower level of evidence and just live with that. And then maybe later we will fix this problem. So, but the algorithms themselves, you know, there's a large number of these algorithms. As you can see, they start with a variant. And by now, there's about 400 different algorithms that predict the impact of a variant. And they give a score that could be between no impact and impact. And that score is, can be a number between 0 and 1, can be a number between minus infinity and plus infinity. Every algorithm is different. There can be a prediction of maybe whether a variant is pathogenic or benign. There could be a prediction of stability. There could be a prediction of splicing disruption. There's just a huge number of these tools that are used. And this word cloud is actually from a paper by Lee in Human Genomics last year. So we can use these algorithms, but first of all, the guidelines themselves do not say which tools to use. They do not say how to use them, what score should be used. If an algorithm gives you a score 0.7, what do you do with that? So it was all left to interpretation to clinicians who actually do not have time to read these papers. They do not have time to understand these papers. Were they trained properly? You know, was cross-validation did correctly? What is the tool actually predicting? Is it predicting what the author say it's predicting? You know, there's a lot of different types of issues that occur in this space. So ClinGen, which is a large body of researchers that called Clinical Genome Resource, has a sequence variant interpretation working group, and they spun out a subgroup that at the time was called computational subgroup, and in the future will be called computational working group, which they charged us with a particular task. So first they said, well, why don't we try to define what it means that the prediction is clinically useful? What do we mean by that? How can anybody claim this is a useful prediction or a tool? They asked us to look at how these tools are used, actually. How do people use tools for the use in the clinic? Do they do it properly? Do they follow what's in the papers? Do they make up something on their own? All of that is true. And then can we develop a solid framework, computational rigorous framework, to actually make some sense of these predictions and give guidance to the clinicians how to use these tools with certain guarantees? So this was a very diverse group, from clinical people to computational biologists to computer scientists. We worked maybe more than 2 years, maybe even 3 years. We did not understand each other very well. We did not speak the same language. I yesterday spoke to Jinwoo, and I mentioned that just the term likelihood ratio, we did not have the same interpretation, what it meant. It took us a year to understand we were not speaking about the same thing. It took us huge headaches. And in the end, we somehow came together and figured out the problems, and I think came up with a solution that I want to present. The first thing we discovered is that we looked a little bit back to find the first tool that was ever developed to predict impact of variants on protein function. And we found this paper from the very first ISMB conference in 1993 by Larry Hunter and Terry Klein. They had a paper that predicts lethal from non-lethal variants in osteogenesis imperfecta. And their data set was printed in the paper. The data set is here on the left-hand side. These are all mutations to alanine, all mutations to serine, and so on. And the model is on the right-hand side. So everything here, there's no deposition of data as we do today. Everything was printed, and even the sequence of collagen was printed in the paper. So that is the beginning. Now we have the word cloud of 400 different tools. I will think of them as binary classifiers. They predict some kind of pathogenic versus benign. And then we found that our goal should be to map these scores, to segment this score range into groups that are compatible with ACMG-AMP guidelines. For example, we need to have supporting, moderate, strong, very strong evidence for pathogenicity and benignity, and we have to have some kind of range in between that we will call indeterminate. So we call this calibration, and I will show you how this works. So I am going to show you a couple of formulas. I will not dwell on them, but I think it's important to note that this has to be a probabilistic framework, and the very guidelines by ACMG and AMP have defined a variant to be likely pathogenic if the certainty is 90% or higher. So that gave us a foot in the door to use probabilities here. And if you see all the way on the right-hand side, probability of pathogenicity given evidence, if it's greater than 99, that's a pathogenic variant. If it's greater than 0.9, it's likely pathogenic. And then similarly for benign, this was actually not defined as 1% and 10, as one would think, but they defined it originally as 0.1% and 10, which has later changed to 1 and 10. And everything in between was variants of uncertain significance. So we started with a basic statistical formula, which said that the posterior odds equal prior odds times the likelihood ratio. So just a brief explanation of what I'm talking about here is that if Y is a random variable, meaning pathogenicity, and E is random variable, meaning evidence, then the thing on the right-hand side is the posterior odds. So basically posterior probability divided by 1 minus that. Everything on the far right-hand side is the prior odds. So we need to know the prior probability that the variant is pathogenic. And then the first term on the right-hand side is called likelihood ratio. I will show you what that is. So the modeling that we undertook was first proposed by Sean Tavtigian and others, who modeled likelihood ratio using an exponential formula where there's one parameter C. And then in the exponent is the number of supporting lines of evidence divided by 8 plus number of moderate lines of evidence divided by 4, number of strong lines of evidence divided by 2, and number of very strong lines of evidence divided by 1. What you can see in that formula is that one line of very strong evidence equals the 2 lines of strong evidence. So there's exponential growth, equals 4 lines of moderate evidence, and equals 8 lines of supporting evidence. So because the evidence is additive here, you can show easily, I will not show you that here, is a naive Bayes model, some kind of reduced naive Bayes model with single parameter. And if you know the prior probability of pathogenicity, it's super important to estimate that prior, then you can find this constant C such that all the rules, all the 20-some rules in the Richards paper from 2015 are satisfied. And they did not know what the prior should be. They decided we will take it to be 10%. They said by experience. And under those conditions, C was 350. And what is really likelihood ratio? Likelihood ratio, if it's a binary predictor, for example, and if you binarize the scores of that predictor, it simply ends up being the ratio of true positive rate and false positive rate. So if you have your predictor, you know what the likelihood ratio is. Now, this is for discrete evidence, and we had to transform that to work for continuous evidence. And this does not have to be just for predictors. It could be for any types of evidence that is continuous. So what we decided is to modify this formula and to compute the local posterior, so local posterior odds, probability that there is disease, given a score S, a continuous score S. And we had to modify the likelihood ratio to become a density ratio. So without much explanation, I would just say that that density ratio ends up being the slope on the ROC curve. Okay, for the math, you can see the paper. But so that is our contribution, how to estimate correctly the posterior probability, the local posterior probability given the score, and part of that was to estimate this density ratio. That density ratio can be estimated fairly easily if you have enough data by a sliding window approach. And so if you rank all the variants according to their score, where the red ones are pathogenic and blue ones are benign, you slide a window and you compute simply the number of pathogenic variants divided by a number of pathogenic variants and some kind of weight, plus some kind of weight times the number of benign variants. And this had to be done to adjust for the prior, because the data in ClinVar, where the variants are that are pathogenic and benign, is biased towards pathogenic variants. There's many more pathogenic variants than you would see. The fraction of pathogenic variants is much larger than you would see in NOMAD. And we used algorithms to estimate that prior to be 4% for missense variants. And so now if you estimate that along the score range, we expect a curve that would go like this. This posterior would go up as the score goes up. You know, ideally linearly if you have a great predictor, but generally that's not the case. Then we also expected for benign variants the probability of benignity to go up as the score goes down. And basically these lines would be defined as supporting moderate, strong, and very strong lines of evidence that depend on that framework that I showed you before. And in the end, you have discretized the entire score range into these intervals. So that works, and we wanted to see how well that works. And so we took one of the most popular predictors called REVEL for impact of missense variants. So we only looked at missense variants, and this is the real result from our paper, where you can see that REVEL reaches supporting line of evidence with score about 0.65 or so, moderate at maybe 0.79 or so, and so on. It reaches strong line of evidence in the end. And that's very interesting because the committee has not awarded more than supporting to algorithms. Now we show for the first time that actual algorithms were better than they believed, and that they can be used at a more significant strength. So we have calibrated 13 different tools in this paper, and then in a follow-up paper that just was published this year, we calibrated another 4. And these are the recommendations that from our paper that have been now adopted by ClinGen and will be part, we think, of the next ACMG/ClinGen guidelines. And the most important line is the top line, is that originally algorithms could only provide supporting lines of evidence, and now it can provide strong, moderate, supporting, and other lines of evidence. So we showed that algorithms are better than thought. They can be reliably used in clinical practice. We then looked what would happen to variants, to all of these variants of uncertain significance, if we applied this new system. You know, will we reduce the number of pathogenic variants, or we will increase the number of pathogenic variants? It's not quite clear, because the use of algorithms previously has been very non-standard, and there are a lot of problems with that. Luckily, it turns out that we will reduce the number of VUS variants by about 19%, and this is from the data from a varied curation expert panels, so-called VSEPs, that work under ClinGen. Many of them will stay the same. About 80% will still remain the VUSs, and some small number of them, about 1%, will become likely benign variants. So the system is actually better. We believe it reduces the total number of uncertain variants, which means that it should increase the rates of diagnosis that are given to the patients. That is something we are trying to measure right now. What is the actual impact of this system in the real world? Have we really improved anything? And how do we measure that properly? But before that, we ran this algorithm and our calibration on a number of genomes from the Rare Genomes Project in Anne O'Donnell-Luria's group. And we found that on average, if you look at maybe REVEL as the third tool, on average per genome, one variant is predicted to have PP3 strong evidence. So this calibration does not overpredict. We are not going to flood the world with a lot of predictions. In fact, will be one per genome, and this is only in disease genes. Moderate variants will be around 3 for REVEL. This is the median, and there will be another 3 variants supporting. So less than 10 variants will be potentially playing a role in diagnosing the patients. So that's a good thing. But there is a problem. that I want just to alert you about, something that needs to be fixed in the near future, which is studied here by Doug Fowler's and Leah Starita's group from University of Washington, is they immediately took the results of our paper and they plotted on all the genes, just as we did in our paper. And so this is the Revel score that goes between 0 and 1, and pathogenic and likely pathogenic variants are red and orange, and benign and likely benign are blue and dark blue. And they decided, okay, these guys did a good job. This works as intended. However, what they also found is that it does not work as intended on all genes. So this is one example of a gene, MSH2, where our thresholds for pathogenicity are a little too permissive. Okay, so we are promoting some variants falsely into pathogenic. We're giving them evidence of pathogenicity when it should not be. And of course, there will be other genes where we are too strict. So on average, it works fine, but on each particular gene needs to be verified. And so this is something that we are working right now on, and there's others in the community working on how to perform a gene-based calibration such that these models can be used reliably on the genes of interest for every particular clinician. And my final couple of words is that we also looked at what happens for different populations. It is very important that these algorithms work well on diverse populations. In the United States, we want this to work on different types of ancestries. And so here I'm just showing you results from Biobank this is unpublished results so far, where we just show what is the number of rare missense variants in disease genes in individuals of different ancestries. And so if you look at, let's say, Europeans, which is the brown bar, there will be around 80 or so missense variants that are rare in a person's genome. If you look at African group, that would be the light green one, there's close to 150. So African individuals have roughly double number of rare missense variants as European individuals. And the question will be, how do we ensure that they are all treated properly in the medical system if we use algorithms or any other types of evidence? And what is fair in this case, when one group has more variants than the other, and other ancestries are somewhere in between? And so that's the same picture as on the upper left-hand side. What I want to show you is the lower bar. This is for alpha missense that we also calibrated, that shows that surprisingly, maybe not surprisingly, but definitely good, is that algorithm gives the same median number of predicted variants across all ancestries. We thought this was really, really important so that there's no overprediction for any type of ancestry. Actually, we have some explanation for that, but I do not have time to fully go over this. What it comes down to is that there's more benign variants in certain ancestries where the number of pathogenic variants is roughly the same in all populations. And with this, I have shown you a little bit about how algorithms are currently used in clinical practice, how we have produced a system that we think is better and will lead to better outcomes in the clinic. And evaluation of algorithms is extremely important. Of course, development of algorithms is extremely important, but properly evaluating and delivering those results as recommendations to the clinicians is of supreme importance. They will not have time to read our papers and do a proper job. They have people to cure. So we are the ones who need to deliver on these recommendations and how this will be used. So towards evaluation, I just want to end up with an announcement. I'm also one of the co-chairs of the KG experiment with Stephen Brenner. And we are doing KG goes for critical assessment of genome interpretation. So we evaluate algorithms in many different scenarios, and KG7 will happen this summer. So I hope that you can participate and test your algorithms on real life problems in KG. And so with this, I would like to thank you for your attention.
Thank you, Fraser, for your wonderful talk. So due to the limited time, so we don't have a question session. So let's move on to the next speaker.
I know, but I gotta hook up my computer. How do I do that?
Thank you, Professor Nam. While the presenter is preparing the computer, And let me introduce Professor Sun Kim again, as he will chair the second keynote talk.
All right, this is my great pleasure, honor to introduce Professor Bonnie Berger at MIT for this talk. And she has so many things, so I'm going to make it short. And she is a member of National Academy of Science, United States, and then also a member of American Academy of Science. And then also, she got the ISCB Senior Scientist Award, and then many awards. And then also, she's actually Simon Professor at MIT, head of the computational biology group, and C-Shell lab at MIT. And then she was RECOM steering committee chair for 10 years. And then, surprisingly, I found out that she's the very first person to do the large language model for proteins. And then also, she is the one of the first person to identify the importance of pairwise residue interaction, which is critical information for hyperpolarized structure predictions, actually. Okay, welcome Bonnie for her talk.
Thank you. Hi. I'm looking for a laser pointer, which was supposed to be up here, but it's not here. Okay, hi. Good. So this works. I don't even know how this works. Anyway, hi. So much of my work has centered around transforming and creating ideas that are from algorithmic thinking and machine learning to biology and biomedicine. Today I'll focus on challenges and opportunities for secure and collaborative drug discovery in the age of generative AI. And Sun Kim said a little bit about this. So this won't advance, and I don't understand why. My own computer won't advance. I need help. I can't advance the slide. I can't advance the slide with my own computer hitting enter. Am I supposed to hit here? It won't even let me go to the next slide. Sorry. What are you doing? Okay, does that work? That should work. Oh wait, let's try. Yes, sorry. Anyway, I'm going to start talking. So, developing a new drug takes years of research and tremendous resources, as we all know. And I'm going to show you a plot right now, which should show up there, but it's not. Okay, here we go. Okay, on the left here, you see the amount of inflation-adjusted money spent by pharma on developing new drugs. And on the right, you see the number of new drugs. Now, the thing is in the last 30 years, in the midst of the genomics and also computer chip revolution, we've spent 6x increase on developing drugs, but we've only approved, at least in the US and similarly in other places, around 2x. I pray to God this goes. Oh, okay, good. Okay, so, and this is not working for some reason. I don't know why. Okay, so the typical drug needs about a decade and $1 to $2 billion to bring to market. So, and this is just background on the problem. So the primary reason for this is the drug discovery pipeline. And as you can see outlined in there, there's a funnel. So what happens is, when you have a new drug candidate, it goes through lots of rounds of evaluation, as you know, and then it finally reaches clinical trials, which are phase 1 through phase 3, And as you can see, we start with over 10,000 candidates here and end up with, 6 candidates, 4 candidates, and 2 candidates. Now, if by chance we could come up with better ways of prioritizing drugs at the top of this funnel, then perhaps we can do better further along the pipeline and have a lower rejection rate. And this would make drug discovery cheaper and faster. Because sometimes a drug may fail later on down the pipeline because it may be like toxic, or it may not be as effective as we would have liked it to be. So a case in point is the drug-target interaction problem, and that is to predict if a small molecule drug, which you see on the left, will bind with your protein of interest, which you see in green on the right. So you can also represent these by, on the left, a SMILES string for the drug molecule, and lower down underneath is the protein sequence representation. So it doesn't work. Okay. But this, okay. Okay, scrolling is which one on here? This one? Okay. Okay, here we go. Okay, so if we can do this well, if we can predict drug-target interactions well, starting just from protein sequence, then we can more efficiently screen new molecules. So we can start with those approved by the United States Food and Drug Administration. There are now around 5,000 of those. And there are a subset of those in Drug Bank, a high-quality drug database. And there are around 12,000 there. And as we grow more and more through these databases, we get to the zinc database with at least 200 million. And just think about it. If we have a new compound of interest, then it's going to take, hundreds of billions of combinations to try if we tried each one with the number of, let's say, genes in the human proteome. And that's about 20,000. And there are larger drug databases, like Enamine with 40 billion. So this is a huge problem. And we want to try to solve it. So there are approaches to predicting drug-target interactions, and they typically have a trade-off of speed and accuracy. So on the x-axis, you see increasing accuracy. On the y, you see increasing speed. And at the bottom right there, you have laboratory experiments, which are typically the very most accurate, but they take a long time. And then at the upper left, on the other extreme, you have sequence-based methods, which are super fast, but not necessarily so accurate. So what if we could come up with an AI method that achieved both accuracy and speed? And that's what I'll tell you about first. One challenge we face is that currently all these drugs are scattered all around the world in isolated silos. And for our purposes, protein sequence data is what we're talking about, and drug data. So if we want, especially in the age of generative AI, for example, large language models, to work well, we want to be able to train on larger data sets. And this is what will enable us to uncover truly novel biology. The interaction data is also scattered all around the world, and that's considered ground truth data of what drugs bind with what protein targets. And we don't have a lot of that, is the problem. So that makes language models really handy, because they are very good when you have lots of, types of data, but you don't have a lot of ground truth data. So obstacles to this, as I just said, are lack of training data, that is ground truth data, lack of compute potentially, these huge GPUs, the right AI architectures, as well as privacy and business concerns. So as I'm going to talk about, we are going to develop large language models for drug screening and tell you how they can be used in secure data collaboration. And that's going to be essential, as we will see. The thing about large language models that makes them so good is they capture the distributional properties of lots of sequence databases that are out there really well. So we have a very rich model. And that's going to allow us to do ultra-high throughput virtual screening so that we can get good, eliminate the bad candidates at the beginning of the funnel. So the method that we came up with, complex, that was the first to the best of my knowledge, introduce in iClear 2022 protein language models to virtual drug screening. And as you can see, it answers our need of being fast and quite accurate. And it's able to screen drug target interactions quickly and accurately at the top of the funnel. It's also a lightweight problem formulation, which is really important because these large language models can have tons and tons of parameters and take forever. So by keeping it lightweight is essential. And this is what's going to enable us to do high throughput inference via efficiency and compactness. So here's what our language model is given. Whoops, I advanced by accident. Okay, so we have a protein sequence on the left there. We have a drug smiles representation, which is like a sequence on the right. And that's what our model is going to take in. It's not going to take any structural data, although I think Martin's fold-seq type data would be very helpful in this case. And we actually did use it to predict protein-protein interactions in TT3D. So I think that would help, and we should add it to the model. But this model is just a sequence model. And we want to know if the target protein binds the drug ligand and forms a complex. And this is different than if we did a molecular dynamics simulation, which I'm animating there on the right, because we're just starting some sequence and using a large language model. And it's going to be a much better model in that we ran a case study and we were able to evaluate over 20 billion candidate pairs of drugs and target proteins in just over a day on a single GPU. And we also validated some of those. I'll tell you about it. Okay, so if we want to predict drug target interactions, we want to learn a shared representation of drugs and targets so that compatible ones are nearby in the vector space, which Sun talked about. We can basically represent everything as a feature vector space in generative AI. So we have there on the upper part our pre-trained protein language model in green. And we're going to reduce that, and it's a very rich model, so it captures a lot, which is going to really help us. So we reduce that to a given dimension in pink, D, and then we take a circular fingerprint representation of our smile string, and we reduce that to the same dimension, D, in pink, in this simple two-layer network. And what we want is what's on the right there. We want that the drug in blue and the target in green, that their vectors are close by cosine distance theta there in vector space. So another idea we had was perhaps we could include model specificity by including negative examples. And we did this by using what we call contrastive learning, which was a known concept, but it had not been used in biology at the time because it was NeurIPS 2022. And we used that to distinguish drugs from decoys. So that's how we're going to handle negative examples as well, because it's going to make our model better. So how did we do it? Well, we use negative examples to get an embedding space that is more meaningful. So if you look at the top line there and the bottom line, it's just two different, evaluations once we've trained our model, or while we're training our model, essentially. So you have a target, a drug, and a decoy, and the decoys are the negative examples. Those are things that look like the drugs but don't bind. And on the top line, we get a very good prediction. Our decoy is far away from the margin around the target, but the drug and the target protein are the same. Okay, so we do nothing there. We don't need to penalize in the loss function of our generative AI system. But on the bottom, the decoy is within that margin, so we need to penalize for that when we're training our model. So we apply a contrastive penalty to that. And the final paper for this was in PNAS 2023. So it does really well compared to other sequence-based methods in terms of the area under the precision recall curve, AUPRC, on 3 well-known data sets, testing data sets for drug-target interactions. But where it does especially well is when unseen drugs are not in our training set. They're just completely left out, even ones similar to drugs in our trainer set. And when unseen targets are not, those are the ones not in our training set. So it enables better performance on drugs or proteins that are fully held out during training. So it's nice when other groups validate your work. And another group looked at structure-based prediction methods, and theirs is the one in pink. It was Jianping's group, KDB-NET. And they found that our method, complex, shown in gray here, performed nearly as well as their method and better than all the other structure-based prediction methods, which ours was not, on different data sets in an independent study. And we use something which is one of my favorite things in the world, these commercial research organizations, and we use them during COVID as well. And we use them to validate our top scoring small molecule compounds on the left and the protein targets at the top of this. And the amazing thing is that for 2/3 of our predictions, we got a high hit rate, and many with nanomolar binding affinity, which is remarkable. And this was all tested at the commercial research organization. We sent them our chemical, they had their chips of their proteins, and they just gave us the binding affinity, and that really enhanced the paper. Okay, so we've talked about large language models for drug screening. Now I'm going to talk about how we can securely collaborate between all these pharma so that they can share their drugs, their targets, their interactions in a secure way so that they have confidence that there is trust. And they're going to actually be able to predict new drug targets together from all this additional information. And I want to do a call out to Ha-Young-hoon Cho, who's now at Yale University and is from South Korea, and was formerly my grad student at MIT. And he'll be here for the Recom conference, which starts tomorrow. Okay, so failed with declining productivity, drug companies became open to a kind of crowdsourcing approach. And one thing used blockchain, although that was not secure. I could break it through randomization. And there were these efforts by these farmer groups to combine and promote drug discovery. They say with AI, but there wasn't much AI. And we've been talking a lot with Sanofi, and the extent of these collaborations has been limited due to conflicting financial interests and also lack of provability that these systems were truly safe. So the first solution we came up with, which was in Science in 2018, where Ha Young-hoon was a co-first author, and Brian He, who's at Stanford. And what we did was we used what's called secure multi-party computation. So this enabled data analysis while keeping all the data private. So we have companies that have their private pharmacological data. They secret share them with different computing entities. And those are all completely encrypted. All that data is encrypted. And together, they train predictive models of drug-target interactions, like the one for complex that I just showed you. But in the Science paper, it was a while back in 2018, and we just used a simple neural network for that. And then we came up with novel predictions, and our predictive models, including the parameters that they used, and our novel predictions were all provably secure. No one could see them. So our secure DTI model, the nice thing about it, compared to matrix factorization methods and diffusion methods that people had used before for drug-target interaction, those took polynomial time, which was way too many if we're talking about 400 billion combinations. And ours took linear time. And I'll get to that in a minute. We had this simple neural network with 200 nodes per layer and 2 layers. And at the time, it was a novelty that we realized you could use ReLU activation, because that required only a single secure comparison. And we also used hinge loss. They're both now common, but at the time, they made our network very efficient, because hinge loss also required only a single comparison. So we fed in on the bottom our proteins, our drugs on the top, and we ran them through this network, and the drugs, targets, and everything going on in the network was kept completely secure. No one could unravel it. It was provably secure from a math point of view, and I'm a math professor. Okay, and it turns out that we got to claim in Science, so it must be true, that this was the first demonstration of a secure neural net on a large-scale, real-world problem. Not just drug-target interaction, on anything, because we actually demonstrated it, as I'll show you next, on the Stitch database, which isn't as reliable as DrugBank, but it provided size at the time. It had 1.5 million interactions. And by training and testing, we showed that we could predict these extremely well. Okay, so, and secure DTI also outperformed existing plain text methods, all the best ones that were known, some of which used artificial intelligence. But the key thing is our framework was secure, and it outperformed these. And security takes a lot of effort. And we tested this first on DrugBank, and it blew them out of the water on Stitch because you couldn't even run any of them. The only ones you could run were matrix factorization and collaborative matrix factorization, and they had an area under the precision recall curve of like 0.5 or less, and we got 0.95. So, and this was on Stitch, so it had extremely good performance. So, in summary for this model, we start at the bottom with different pharma companies with their privately observed drug-target interactions, and then they collaborate. In the first step, on the left, they secret share, their data as much as they want, and we pool those data sets, and the colors correspond to the data sets for each of the companies. And then we perform our secure computation framework over those pooled data sets at the top, number 2, and we use them to jointly train predictive models. And these can be large language models, even though ours was simpler at the time, and I'm going to get into that in a second, because we've actually gotten that to work, but it's not published yet. And they predict novel DTIs, which are also encrypted, and then they distribute the results. And each of the entities can decrypt their results, but they're distributed in a way that none of the other entities know anything about the underlying drugs, protein targets, or the interactions in a provably secure way. Now, of course, everyone knows their protein targets, so there are 20,000 of them, so they may have a hunch about those, but not other things. So, what we're working on now is, in the age of generative AI, we want to do secure and collaborative drug discovery for more complicated foundation models. So, for instance, like complex that I showed you earlier. And this is just a pictorial of complex. But now, what we want to do is we want to lock or encrypt the protein foundation model for the upper protein and also the circular fingerprints for the drug, so you can't identify them. And lightweight problem formulations, such as we had for complex, are going to be essential in deploying privacy-enhancing technologies. So here's what our work is cut out for us, and we've done some of these steps and not others. So here's what we require. We require private and secure language model inference. That is the one part we figured out. Then we also require secure parameter-efficient fine-tuning. And we just had a paper in 2024 in PNAS with my student Sam Sledieski. And here, what we do is, if you look at the bottom, that's what happens when a classifier head is from a multi-layer perceptron. That's the standard way of doing things. You have to train the whole darn thing, all the parameters, and then you get these weights that are updated, and you keep retraining. But in parameter-efficient fine-tuning at the top, like the LoRa algorithm, you only have to train the heads that are making a difference. And that sparsifies the number of parameters, and turns out it's more accurate because you zero in on what really matters. So we're also applying that to make lighter weight models, and we have done that successfully for protein-protein interactions already. But here, the key is, we want our data to be encrypted. We want our attention head data to fully be encrypted. And we are able to do that. So the last step is we want to be able to share these adapter weights trained by the different computing parties for lightweight multi-party homomorphic encryption. So we have these adapter weights. We want to share them to train our pooled model from the different sources. So we need them to be encrypted. And we have a very good way to do that. And And one recent approach, which is we're using actually for this, but we've used it in a different context. And the context is that we came up with a cryptographic approach with J.P. Hubert, who is at EPFL, and his former student, who was my postdoc, David Froelicher, and Hayoung-Hoon Cho. And it just appeared in Nature Genetics in 2025, and we're using a very similar approach for our secure drug target interaction. And so remember, secure multi-party computation was conventionally based on secret sharing, and there are a number of references here for secret sharing. But that meant that all private data had to be shared securely among the parties. And that's just way too much data. All your private data for everything, there was just too much communication going back and forth in the cryptographic model, and it couldn't scale necessarily beyond 1.5 million, which we did test on. But we're now talking about hundreds of billions. So we came up with this model called multi-party homomorphic encryption. So what that allowed you to do is it used multi-party computation, secure multi-party computation, but everyone kept local data unencrypted. That was their own personal data, like the pharma companies. And only intermediate results were encrypted and shared in a multi-party secure computation framework. And basically, they did homomorphic encryption locally. And then we got output that was distributed to either of, let's say, two entities that were communicating. And only the final output is revealed. The intermediate results are completely encrypted. And we came up with distributed algorithms, so a lot of the computation could be done locally with homomorphic encryption. And then, so our question is, can multi-party homomorphic encryption, our new approach schemes, be applied to address the costs of large-scale generative AI models? We were able to already use it in our PNAS 2025 paper and in our Nature Genetics 2025 paper, sorry, to answer GWAS questions. That is, you want to identify genetic variants linked to disease. These two genomes differentiate in one position, a SNP, and given an individual genome, we want to know how does it differ from others. And those are called genetic variants, and Pedro was talking some about those. And there's a whole complicated formula with, is a genotype, the genetic variant, related to the phenotype, where you also include covariates like age and sex and environmental noise. And we were able to do this on a large and diverse study of cohorts. And that proved to be necessary for more accurate analysis, as Pedja alluded to. So we can do this in a totally provably secure way using multi-party homomorphic encryption, which we call secure federated GWAS. And that enabled large-scale genetic studies across siloed data sets. So we can get tons and tons of more data for fairer representation of the data set, of individuals. And I'm not going to go into this now because of time, but we actually did it for UK Biobank, all of us in the US, a huge database, and the Millions Veterans Project in the US. So we did it for millions and millions of GWAS studies. And we have full software in this paper, which I forgot to add at the end because it was just out. So another way to make these models lightweight is to use what's called sketching. You can sketch your original data that's similar to each other so that you don't have to run on all that data. And we came up with this particularly with compressive genomics in 2010 or 2012 in Nature Biotech, and then in Cell Systems, we developed a theory for it where, because of evolution, biological data and even drug data is actually quite similar to each other. So it can be projected onto a low-dimensional manifold. This is just in 2D, but where you can cover it with these equal-sized spheres, which can be clusters, where you take a cluster representative for each, and you don't need the other things. And this is lossless compression. So we wanted to use ideas like this to make a general framework, to make privacy-enhancing technologies and multi-party homomorphic encryption practical for drug discovery. And we were able to do this on a genetics problem, which actually last year my students Matthew Hung and David Froelich, postdoc, got the Best Student and Young Scientist Paper Award at RECOM for this secure relative detection paper. So here the problem we're interested in is there a third degree or closer relative in the data set. And we want to do this in a secure way. And it turns out that we can actually detect third degree relatives. And that wasn't the secure part, but we can also add security on top of that. And the way we did it was we sketched the genome to of each individual, to group individuals for comparisons. And then we didn't have to make as many comparisons because we were using sketching to figure out if there were these third degree relatives. And the key thing here is that by doing this, you could see that you could do this possibly with drug-target interactions if you sketched what drugs and what targets tended to interact. So this was published in Genome Research, which had a partnership with RECOM, has a partnership with RECOM, and so does Cell Systems. So that problem is essentially in one database, in another, are there relatives, third degree relatives? And the reason you'd want to know that is because, well, you might want to find them, but the reason you want to know that is it can distort your findings. Like you need to know where the relatives are. Okay, so food for thought. We might want to return the results, that is the trained drug-target interaction models and or the predictions in a way that benefits the participants fairly. Meaning if you're Sanofi and you've put in a ton of data into this collaboration, you and someone else has put in just a little bit, you should get more out of it. So that's one thing. And we also have the need for foundational frameworks that can support the secure training of a variety of predictive models, different protein language models, different types of predicted models, and we need to be able to adapt them to these privacy-preserving frameworks, or as we say, PETs, privacy-enhancing technologies. And Ha-Young-hoon and I are working a lot on that. And then there's a huge role for government and policies to lead initiatives in encouraging such partnerships between pharma. So here's just some of the software. The main software that was for Complex in the first part of the talk, and Secure DTI on the bottom. There's other software that you can find in the papers. And I want to thank, in the top row, all those folks, Rohit Singh, who's now at Duke, who is my grad student at MIT, Slam Sudeski, my grad student at MIT, who's now actually at Simon's Flatiron as a postdoc, Brian Bryson, my amazing collaborator in bioengineering, he's a professor at MIT, and Lenore Cowan, my long-term collaborator at Tufts, who is a genius. Anyway, and then there's Young-Hoon Cho, who I spoke about, and Brian He, who started with us on the science paper in 2018, and he's now an assistant professor at Stanford, and Ha-Young-hoon is assistant professor at Yale, and Rohit is an assistant professor at Duke. Anyway, thank you.
Thank you, Bonnie, for sharing your recent work in interesting and exciting ways. And then, because in the interest of time, we don't have question. However, you can use lunch break, and then we can ask question, discussion while recent talks. Thank you very much.
Thank you very much, all speakers and chairs. We will now break for lunch. Lunch will be served here at this place. And additionally, the banquet at the end of today's program will also take place here. So please enjoy the meal and networking opportunities. The program will resume at 1 o'clock p.m. Thank you. The lunch will be served right here, so they will bring you lunch in your place, so please be seated for your lunch. 점심 식사는 앉아 계신 테이블로 가져다 드릴 겁니다. Hello everyone. We will soon start our afternoon session, so please be seated. Welcome back everyone. Now we move to scientific session 1. To chair this session, please welcome back Professor Sang-ho Ryu, President of AIxBIO.
Hi. So how was your lunch? Was it okay? So the dinner will waiting for us, and dinner is much better than lunch, I think. So hopefully you can enjoy the dinner also. So this session, we have three distinguished professors from all over the world, and I'll be expecting very nice talk today. And let me introduce from the first speakers. The first speaker is Professor Jae-woo Kang, and he graduated Korea University as a bachelor, and after that did a master's degree at University of Colorado, and after that he got a PhD at University of Wisconsin Madison. After that, he has some experience in AT&T lab research, and also did an assistant professor at North Carolina State University. After that, he's serving as a professor at Korea University. Also, he's a founder of AI Gen Sciences. Also, he's very actively working on the industry and academics. So please welcome Professor Jae-woo Kang.
Thank you very much for the introduction. I'm honored to be here to share some of my story in my lab and also in my company. Again, I'm a professor of computer science at Korea University and a CEO of AIxBIO Sciences. And AIxBIO Science is a spin-out company from my lab at Korea University. We are applying AI to develop the drugs. And so we are doing full-stack AI for drug development. But today, I'm going to focus on the LLM side of the story. So I'll start with the BioBert, which is the language model that we developed in my lab, and that happened to be the first and the most cited biomedical language model. And then move on to the Meerkat, which also happened to be the first small language model that passes US medical license exam. And finally, I'm going to introduce briefly how these technologies apply to the actual drug development. Like you all know, the drug development process starts from the target identification. Like all science, drug development also starts from the hypothesis generation. The most important hypothesis in drug development is target hypothesis. So which protein or gene, by designing a drug that can inhibit the protein target, that can suppress the mechanism of disease. That's the target hypothesis. Once we develop the target ID, then we design the drug that can bind to the target, and then test on the animal, preclinical stage, and then move on to the human clinical trial. This process typically takes 10 to 15 years and costs more than $2 billion US. And failure in the later stage of drug development is very critical. It's almost threatening the survival of the company because of the cost of the R&D. And one analysis reports that the clinical failure causes well over the 50% of the cause of the clinical failure has to do with the hypothesis, the wrong hypothesis, which is the wrong target selection in this case. So target selection is very important. So in order to do the target hypothesis generation, we need to understand the mechanism of disease and also have to do the hypothesizing target gene or protein that capable of deactivating the mechanism of disease. And it typically starts by searching academic literature. The problem is that you all know PubMed, right? It has more than 37 million articles indexed. And even more troubling fact is that more than 3,000 new papers are added every day. So it's all peer-reviewed journal articles, more than 3,000 every day. So it's way beyond the human capability of the manual curation. So we need help from the systematic approach, like using AI. So we wanted to develop the language model that understands the professional language like biomedical literature. So it all started from this event, the Multi-Targeting Drug Dream Challenge, happened in the end of year 2018. So we attended this challenge and won the event, and so we attended the award-winning award ceremony at New York. After the ceremony, I gathered together with my team and then had the beer. And Jinhyuk Lee, who is now at Google DeepMind, said that, Professor, the BERT is SOTA these days. So I goes, what is BERT? So it's a language model that understands human language. So what does it do? So it does all those different tests and then dominate the previous SOTA models, right? Including name entity recognition and relation extraction and question answering. So the BERT turns out to be the pre-trained encoder of transformer architecture. So transformer architecture has an encoder-decoder. And BERT is an encoder-only model that's pre-trained on the Wikipedia corpus, primarily. Right? So it seems to understand the general human language, like a Wikipedia level. But I got curious whether this model can be trained to understand the professional language like biomedical literature. So, my team and me went back to school and then work on do the continual pre-training on this BERT model. BERT is pre-trained on the Wikipedia and get this BERT pre-trained model and then continual pre-training on the biomedical literature. So original BERT was trained on the 3.3 billion words, and we trained on the PubMed and the PNC full text, which consists of about 18 billion words, about 6 times larger than the original corpus the BERT trained on. So it took about 20 days using 8 V100 GPUs. And we tested on the key downstream tasks which are essential building block to build a machine that understands and systematically process biomedical literature, which is name entity recognition, relation extraction, and question answering. And surprisingly, this one single model dominates most of the previous SOTA in most of the benchmarks. So one of the examples is question answering. There is a challenge called BioAIxBIO, which is a Google-sponsored challenge. It's like the given literature and then the AI trying to answer the question about the literature. Right? That's the challenge. And the 4th and 5th and 6th challenge, those are benchmark data. We use those benchmark data and evaluate our BioBERT model. And it happened to be, it beats all the previous winning model in the events. And so we archived the paper in January 2019. It's been only like five weeks after we initiated the discussion about building this model and came back to Korea. And we worked on the 5 weeks over the Christmas holiday and archived paper. This happened to be the paper written the shortest time in my lab, and then happened to be the one that has the most citation. And we figured that the BioAIxBIO 7B challenge just about to open in 1 month after we archived the paper. So we decided to participate in the challenge. And the challenge is like this: given the part of the literature and then question, and the AIs, participating AI models supposed to get the answer and then report to the answer. And AIs takes the 5 exams, the 5 different batches over the 2 months period. And then all the competing models like SBM, LSTM, the conventional machine learning models. And we are the only team that brings a language model to participate and compete in the challenge. The result was quite surprising. All 5 batch, we won the first place. And then Google takes the second place in the 4th and 5th batch. And our model gets, the feasibility after this, the winning of this event. So get some citation. And the next year, we also wanted to participate in the same event. Because we opened the model and the trained the base of the BioBert, all the competing teams in the next year's event started from the BioBert. So we had to do some improvement in order to keep the winning position. And we managed to the win again. So 2 wins in a row in BioSQ. And the citation number is a little bit outdated. Today it's about like 7,000, slightly more than 7,000 citations. The competing models include the Microsoft PubMed BERT and NVIDIA BioMegatron, and AI2 Cybert, etc. And those 2 events that I just explained is 2019 7B, 2020 8B BioSQ challenge. And we also participate in different challenges. Those are the question answering, and there are another challenge called BioCreative 7, which is named entity recognition and relation extraction challenge. So we teamed up with AstraZeneca, and as a one team and participate in this challenge, and then we achieved another the win in that event. And then we developed the vision language model to participate in the radiology image diagnosis challenge and compete with the Siemens. And then we won the event. And the most recent challenges are about the text mining or natural language processing in biomedical domain. We also doing some drug discovery, like a drug design or drug screening challenge as well, including AstraZeneca Sanger Dream Challenge and multi-targeting drug dream challenge, etc. But today, since the topic is about language models, so we are going to focus on this part. So after the BioBert, that's the encode-only model, right? That's capable of understanding the professional language like biomedical literature, but that's encode-only, so it's not capable of generation, so text generation. So we move on to the decode-only model. So most of the GPT, the language model these days are decode-only architecture. So we want to build a biomedical language model that are capable of generating some professional text and understand and capable of doing the biomedical reasoning. So we developed Meerkat, which happens to be the small language model that first passed the United States medical license exam. This is just got accepted and then is impressed in MPJ, digital medicine. The process goes like getting, just like we did the BioBert, we get the BERT, and then continue pre-training and do the fine-tuning. We get the foundation model like Llama and Mistral. We trained on the general text, and then continue pre-training on the biomedical specific corpus, and then generate multi-hub reasoning examples and fine-tune this pre-trained model, and then we get the Meerkat. But this process takes a lot of resources and time, so we didn't do it at this time, but in the future we may do that, but for now we only do the multi-hub reasoning, fine-tuning. So because it is a small model, like a 7 billion parameter, which is very small, that can fit in just one single computer. The conventional next token prediction autoregressive pre-training is not working. We tried it, but it didn't work. So we get the high quality text, which will be the medical textbook. So we collect the 18 medical textbook. All the medical school students in the US study to pass the exam. So we get the 18 textbook. And then read one word by one. But you know, the small language model does not has the capability of the complex reasoning. It didn't emerge. So we design a different way to train the small language model. So a little more the AI-friendly way to train the model. So we generate the US MLE style questions from the chunk of the textbook, and then generate a chain of thought reasoning process to solve this question one step by one. And then this problem and the chain of thought data together to fine-tune the language model. Like this, from this textbook, this chunk of the textbook, generate the USML style question, and then generate a chain of thought, step-by-step reasoning to solve this question. Okay? And then problem and the question pair, we do the fine-tuning. And that was quite successful. The same size model, the previous SOTA was Meditron developed by EPFL, and they achieved like 52 points. But our model, using this way of AI-friendly the training method, is achieved a 77.1 score. So on average, USMLE passing score is like 60 points. So it's well beyond the passing mark. So we have the 10 times bigger model, 70 billion, and this achieves like 85.8, which is a quite high score. And not only just solving the USMLE style question, so it's capable of a variety of tasks. So one example is that the NEJM case challenge, which is you know, like a 2-page patient description is multimodal. It's like a text description including like the lab data, as well as some images, pathology images. And there are 6 options, multiple choice question with the 6 options. And those are the score distribution of the human experts. So there are about 38 questions. Because it's a multiple choice with 6 options, by guessing, you achieve slightly more than 6 questions. You get 6 options, right? But the human average is about 11 to 12 questions. The human expert reading NEJM. But GPT-4 at that time achieved 21.8 questions correctly answered, which is about 99.8% percentile of human experts, which is already very capable model, right? But it's a huge model like GPT-4. But small model like ours it can perform. So we tested it on this NEJM challenge, and it's achieved the 21 parameters correctly answered. It's very close to the GPT-4. What this signifies? So it's a small model that can fit into one small computer, one server. So any institution who are interested in applying this, the biomedical domain expert LLM to process their data, they can do that, you know. So deploy on-premise in their local, you know, server. So this is a paper, and this is the AI-generated meerkat image. So, you know, there's the AI has some hallucination issue here. It's a 4, you know, the color, and it's only 3 colors. So we tried to fix it, but didn't understand. So we just keep this image. So not only just question problem solving, so the patient consultation. We use GPT-4 to play as a patient, right? We give one USMLE question to GPT-4, and then based upon this USMLE question, you play as a patient. and consulting a doctor. In this case, the MuirK7B plays as a doctor, right? So this is a doctor played by MuirK7B, and the patient played by GPT-4. GPT-4 only sees the context, which is the USM education. MuirK7B is not seeing that question, doesn't know the context. So in the mother, concerned about 10-year-old son, painless mass in the left testicle. So the MuirK7B playing the doctor, trying to collect the information from the patients, like any other symptoms or medical history. So all the collected information finally said that I will schedule the ultrasound. Okay? Probably the correct next step in this case, in the patient consultation case. The same mother, but this case with some lab test exam data and the biopsy result. So those are the information given in the USM medication, right? So it provide that information together with the symptom. The MuirK7B directly go to the yolk sac tumor, which is the correct diagnosis in this case. So in this case, surprisingly, small mother correctly diagnoses these patient cases. As another example, USMLE, this is the problem-solving process of the MuirK. Each option, the reason that the option is inappropriate, and the reason this option is appropriate, and then, therefore, the answer is B, cholesterol embolization. This is a chain of thought reasoning. And other clinical questions like I have a wisdom teeth removed upper and lower 6 days ago, but the lower, they pulled tooth without removing the root. And I noticed the lump about 1 inch between lower jawline. And I'm taking this medication, so what's the cause? Those, the everyday, the clinical questions also capable of answer the question. So after this, and then we got curious whether we only trained using the English version of the medical textbook. So we were curious whether this can pass the Korean medical license exam as well, right? The medical knowledge, whether the medical knowledge can translate to the Korean cases as well, right? So this is the Korean medical license exam question, and it answers in English, but the answer correctly. Because we didn't align to answer Korean, so it's still understand the Korean question, but the answer in English. And we give the question translated in English, and it also understand and then correctly answer. The score after translated the question, English version of it's the same question, but the Korean question score is a little lower because it's natural because we didn't align the Korean version. So in KMLE, it achieved 80, which is a quite high score. And we now align to the Korean context to make the MuirK version 2. And now we have more than 90%, 90 point, slightly more than 90 point in the Korean medical license exam. So she's a very high score. So in a couple of minutes, I will introduce how we apply this technology to do the real-world drug development. So using the proven AI drug development platform, we designed the drug de novo without drug screening design, and then achieves lead compounds in just 6 months. The conventional, traditionally, it takes about 2 years. We saved about 70% in R&D cost compared to the industry. So, and using this technology, we develop multiple different pipelines, mostly the oncology drugs, and also develop ADC drugs as well. And combining those, the founding, the award-winning technology into the AIxInsight and ChemTeller. AIxInsight is LLM agent, and ChemTeller is also LLM agent, but specialized to the drug design. And Insight, this is a quick demo. Yeah. Trying to generate the hypothesis using the biomedical literature and validate those hypotheses using the experimental data, and also combines the data-driven hypothesis, like differential gene expression analysis, and prioritize those hypotheses using literature evidence. We can combine together. So data-driven hypothesis and literature-driven hypothesis combined together. And validate using some experimental data as well. And then we confirm, we validate one target and get the target, and we start to design the molecule. To design the molecule, we use a chemtailor, which is specialized for drug design. And using a diverse drug design models, with AI precision, just 30 synthesis is sufficient to identify a lead compound. So far, we didn't exceed more than 30 compound synthesis. And then we ID'd the nanomolar scale, the activity lead compounds. So here's the chemtailor example. ID the target and get the target, structural target, and then design a molecule that can fit into the target protein. So this is the process of the chemtailor LLM. Get the structural target, and then identify available tools, and then plan how to design this molecule for this particular target, and then utilize external tools to get the intermediate result, and then pass this intermediate result to the next tool for the analysis, and finally get the structure of the designed molecule. Right? And once the molecule is designed, we validate the activity of this drug. So we have a well-lab facility in-house. The space, we do the in vitro analysis, like a cell culture. And this, the in vivo validation facility, we do the xenograft mouse. We do the mouse test to put the cancer cell into the mouse. It's a xenograft mouse, and then see how the cancer cell grows. And after the treating the drug, how the cancer cell diminishes. We validate the drug using this process. And those are the mouse that we tested. And this is another pipeline, the result of the mouse evaluation. So, so this is the end of the talk. Thank you very much.
Thank you, Professor Jae Kang. And we have some time left. So we're going to take one question from the audience. Any question from audience? If there's no question, maybe you can freely talk to Professor Kang during the short break. So thank you again,
Thank
Professor
you very much.
Kang. Yes. So let me introduce the second speakers, Professor Yu Lee. And Yu Lee got a bachelor degree in bioscience at University of Science and Technology of China. And also after that, he got a PhD in computer science from KAUST. Also after that, he became a professor at Chinese University of Hong Kong, CUHK. Recently he published very interesting papers, Nature, Biotechnology, and Nature Method. Today he's going to talk about complex disease modeling and efficient drug discovery with the large language models. Please welcome Professor Yu Lee.
Okay, thank you very much for the kind introduction. And also thank you a lot for inviting me here. So this is my first time being in Korea. It's always a pleasure to be here. So today I'm going to introduce the recent works in our group. So yeah, so I already introduced myself. So essentially I have a dual background. So I did my bachelor degree in biosciences and my PhD in computer science. And currently I'm in the computer science department at CUHK. So this is the overview of the research in my group. So in the center, seems not working, but anyway, it's fine. So in the center, we're developing AI algorithms, like deep learning algorithms, large language models, and so on. And then we try to resolve the problems in both computational biology and human health. So in terms of computational biology, we're working on sequence analysis, structure prediction, and function prediction. So we can consider them as the foundation and mechanism for the healthcare problem. And then, of course, there are some gap between biology and human health. So we also develop machine learning models to resolve the problems directly related to disease and drug development. So here are some representative works from the group in the past roughly 4 or 5 years. So we develop models to try to model disease and the molecules, like the disease modeling, RNA modeling, and protein modeling. But in addition to that, we're also very interested in developing drug. So we also do the modeling of protein-RNA interaction, as well as to do the design for the peptides and the RNA. So I will try to go through those projects quickly in the next 25 minutes. Okay? So let's start with the disease modeling. So human disease modeling is in fact a very classic problem. So we know diseases definition can be sometimes overlapping with each other, and different diseases can be etiologically linked. But when we go to hospital, we are first be assigned to different specialists, right? They will consider the disease to be different. So as a researcher, we're thinking, is it possible we regard and consider human illness or disease in a homeostatic system or in a holistic system? So this is the thing what we want to do. And in addition to that, we're curious about the genetic parameters estimation. So for example, each disease may have a unique prevalence profile across different patients' age and sex, and also different diseases may have some correlation, and we're also curious about their heritability. So we can definitely estimate those parameters with a huge amount of genetic parameters, but that means we need to collect those data. So we're curious whether we can estimate those genetic parameters efficiently without a large amount of genetic data. So here are some related works. So you can say for the genetic parameter estimation and disease modeling, some of the research can go back to roughly 60 years ago. So it's a very long time ago and a very historical problem. So our solution to this kind of problem is to try to build up a continuous low-dimensional space. So I think for the talks today, so people are already continually talking about the embedding. So I think I don't need to introduce the concept of embedding again here. So we try to build up the representation of the disease and get the embedding, and then do some downstream analysis like the genetic parameter estimation. So what we have done here is to try to collect the data from US, Denmark, Sweden, and use this kind of electronic health records to build up the embedding of different disease. Now after that, we can do lots of downstream things. So due to the limited time, I won't go through the methodology here, but here I want to emphasize the sample size that we collect. So we collaborated with University of Chicago, and we collected a sample of 123 million US people. So it's a huge amount of data. So essentially that's not the classic electronic health records. After all, if you want to have access to almost half of the US people, definitely need lots of time. So what we try to get is the insurance claim data, which you can consider as a kind of EHR. And what we do is that we try to train a model based on the larger language model style training, and then we can map the different disease entity onto a 20-dimensional vector. So why is 20? We tried different until we find that 20 can be efficient and also can give us good results. So although I can't point here, but you can see the figure on the right. So we show those representation of different diseases on the 3-dimensional space. So you can see those different diseases belonging to the same group or with similar symptoms, they are clustered together, which shows that there are some physical meaning of this kind of representation. Okay? And then with this kind of representation, we can build up some very, very simple machine learning model, like lasso or ridge regression or random forest based on the kind of embedding, and estimate the genetic parameters like the heritability, like the correlation between different diseases and so on. So you can see the accuracy of, I would say, like correlation coefficient against the ground truth is quite high, can be as high as 0.9 regarding the correlation coefficient. So notice that here, we didn't use any genetic information. We didn't use any genetic data. We only used the electron health records, build up the model, and then get the embedding, and then build up the downstream machine learning model to do the genetic parameter estimation. And we can achieve very good result. So in addition to that, we can also have some scientific discovery. So for example, to identify some unique genetic loci which were ignored by the previous research and then further validated. Okay? So this is a very brief introduction about how we handle electron health records to build up the representation of the modeling of disease, which can facilitate the downstream genetic parameter estimation and the scientific discovery. Okay? So then let's head to the next part. That is the computational biology part. So I want to start with the Nobel Prize in Physics and Chemistry last year. So we know, so last year is really a year of AR. So AR won the Nobel Prize in Physics last year, and also AR for protein research, such as protein design and protein structure prediction, won the Nobel Prize in Chemistry last year. And we know that for Nobel Prize in Chemistry, the very important model is AlphaFold2. So with this model, given a sequence, we search a database to get multiple sequence alignment, and then put it into a machine learning model, we can predict the 3D structure. So here, I'll put the model here. We can see this model contains essentially two parts. One part is the database searching to get multiple sequence alignment. The second part is the machine learning model. So this model was proposed roughly 4 years ago, and after all this years study, we now know not only the model itself is important, the database searching is also very important because it can give us the co-evolution information from the protein homologs, which essentially serve as a foundation for us to infer the closeness between different amino acids. So this is essentially the co-evolutionary analysis or co-evolutionary assumption. So the observation is that if two residue or two nucleotides within a protein, they tend to contact with each other, then they tend to co-evolve with each other. So here we show the multiple sequence alignment of one sequence, and we find that there are two residues. They are highly co-evolved, and in the 3D space, they are in fact contact with each other. So this is essentially the assumption behind all those models to do the protein structure prediction. So next question is, how do we get those homologs and how to construct the multiple sequence alignments? So here we show some of the foundational methods here. You can see some of the tools are very popular, like StarBlast, right, which is essentially the foundation of the bioinformatics field. We also have some tools developed by my team and also by some Eddie and so on. So those tools are very popular, very famous, and essentially the foundation of bioinformatics. And recently, we also have FoldSeq based on the sequence alignment to find the protein homologs. But those tools are great, okay? But they still have some limitations. First of all, if it's alignment-based methods, then they probably need to balance speed and accuracy. If they run it multiple rounds, they can definitely identify more homologs, but we need to sacrifice the time. It can be very slow, right? And another thing is about the assumption behind those methods. So the assumption behind the alignment-based method is that the homologs are defined by similarity. If two sequences are similar to each other, then we consider as homologs. But this kind of assumption can be wrong sometimes, especially for the remote homologs. So what is remote homologs? So you can see the figure on the right, okay? So those sequences during evolution, sometimes an entire domain can get lost due to evolution, or maybe an insertion of probably a base into the into the DNA, then it can change the protein sequence completely. But from the evolution point of view, we still consider them as homologs. But if we compare the sequence similarity, most likely the sequence similarity can be very low. And this kind of remote homologs will be missed by the similarity-based methods or the lambda-based methods, right? Okay, so this is the definition difference between homologs and similarity, okay? Then what's our solution? Our idea is to try to borrow the idea from the natural language processing field. So if you do the mapping between the natural language processing methods and the bioinformatic methods, you will find that the BLAST is very similar to the idea behind the bag-of-words representation. Hammer is essentially hidden mark model, right? Which is essentially a hidden mark model in the natural language processing field. And our recent method is based on transformer or based on the large language model. And here is our detailed method. So this method is an alignment-free and database-independent homology retrieval method. So given the sequence, we put it into a protein language model to get the representation or get the embedding. And we compare this embedding or do some very simple mathematical calculation against the database. And within that database, we also have the embedding of different sequences of database. And then we can check, maybe perform dot product, maybe you can calculate their Euclidean distance, but no matter what kind of distance metric you are using, it can be very quick. And then you can have the rank. Based on the rank, you can identify those homologs. And with that, you can construct the multiple sequence alignments, and furthermore, you can do the downstream structure prediction. So this kind of dense retrieval can be very scalable, so it can be scaled to a huge amount of sequences. So, of course, for this kind of homolog retrieval, we care about two things. First of all, whether it can indeed retrieve those homologs we care about. And secondly, what's the speed, right? So then let's check the performance. So first of all is sensitivity, whether we can indeed get those homologs back. So we compared our method against all the other methods. You can see Dimer, the Cyblast, Hammer, MM62, FoodSeq, and so on. So which covers the sequence alignment and also structure alignment methods. So compared with them, the performance is similar, which means we are not worse than them. But in addition to that, in terms of speed, so we can increase the speed greatly. So compared to Hammer, we are roughly 100 times faster than them. And also, we can, based on our configuration, we can also accelerate the running time of AlphaFold 2. So, of course, we run lots of experiments due to the limited time. I won't introduce all of them. So in terms of remote homologs, we can indeed recall those remote homologs, and also you can find compared to Cyblast. In terms of the structure similarity, they're very low, but if we retrieve them back using DHR and do the structure similarity calculation, you'll find that we can recall those remote homologs. And the homologs retrieved by our method can also diversify the sequences in the multiple sequence environment put into the protein structure prediction model. So here we finally show whether the model, the sequence retrieved by our method can further improve the AlphaFold prediction. So here we can see we compare the different models, including AlphaFold 2 with only the single sequence input, OmegaFold and ESMFold, which are essentially the protein language model-based methods to do the structure prediction, and also AlphaFold 2 default. So we can see compared with AlphaFold 2, OmegaFold and ESMFold, their performance are good, but not as good as AlphaFold, which shows the importance of multiple sequence alignment and homologs. So the correlation information containing in the multiple sequence alignment is very important. They are still can't be completely replaced by the protein homologs. Protein language models. And if we put the homologs retrieved by the DHR into AlphaFold 2, we can further boost the performance of the protein structure prediction. Okay? Okay, in terms of application, we also modify this model slightly to do the functional retrieval. That is to identify some peptides or proteins with specific function. So here I'll show you as one example. That is, we apply this kind of pipeline to identify novel signal peptides. And we can discover more than 300 novel signal peptides very quickly in roughly just 3 months. So we also have some in-house testing. We find the hit rate is very, very high, roughly 80%. We also applied this kind of pipeline onto other functional peptides and the proteins. So those papers are under revision or under preparation, under submission. So you will see those applications very soon in the upcoming 1 year. Okay? So we know that the academic breakthrough in the protein field has led to substantial transformation. So here I list some of the representative companies and startups here. So you can see the X-Rare, it can even raise $1 billion just for the seed round, right? So in the protein field, this kind of transformation has already been very impressive. So in our group, we have been thinking about what will be the next thing in addition to the protein. So in the protein field, we already have lots of companies over there. So what will be the next thing? So our thought is probably it might be the RNA-based therapeutics. So RNA therapeutics has already been very successful during COVID, you know, the mRNA vaccine. But the mRNA vaccine is just one kind of the RNA-based therapeutics. So if you see the 2 figures here, you will see that mRNA is just one part of it. We also have aptamers, mRNA, siRNA, tRNA, ASO, and siRNA. So those modalities can also potentially be some RNA therapeutics. So we already know it can be highly specific, the production can be quick and scalable, and it can potentially help us to handle some undruggable disease. So in terms of the Nobel Prize, you can see we have a series of Nobel Prize involving RNA. So in fact, for the Nobel Prize last year in medicine or biology, so it's related to microRNA. Although the other 2 Nobel Prize in physics and in chemistry has attracted most of the attention, okay? But there are still some bottlenecks in the computational RNA study. So first of all, in terms of the structure prediction, compared to the protein field, I would say it's roughly 10 years behind the protein field, okay? So first of all, in terms of the available data, for the protein, we have roughly 200K structure resolved in the PD database, while for the RNA, we only have roughly 2,000, okay? If we remove the redundancy, then probably only just slightly more than 1,000 RNA with the structure resolved. On the other hand, for the RNAs, we may not know their function, especially for the non-coding RNAs. They are lacking of annotation. But on the other hand, if we can consider RNA the alternative drug targets, we may have 100 times more targets than the protein, right? And in the RNA central database, we have more than 27 million sequences available. So the idea in our group is that is it possible we take advantage of this huge amount of sequence data to try to push the fundamental research in the RNA field, like the structure prediction, like the function annotation, like RNA design. So our strategy, first of all, is to develop an RNA foundation model, so which is an RNA language model. So we developed this model go back to 2022, okay, a very long time ago. So I think I don't need to introduce this model in detail because it's very similar to ESM, and also very similar to BERT. So given the huge amount of RNA sequence, roughly 23 million, we trained a BERT for the RNA field, and hopefully can push the downstream application like the structure prediction and also function annotation. So we also checked whether this model can learn some meaningful representation or meaningful embedding of the sequence. So here we show some of the result here. You will find that with the RNA-FM embedding, we do the dimension reduction using t-SNE. You can find that in the most left figure, the different RNA types, they can be separated naturally. We even don't need to build up any follow-up machine model. They can be separately almost perfectly, Okay? So which means that RNA-FM can potentially learn the language of RNA and extract the function and the pattern from the sequence. Okay? Then we headed to the next application. That is whether we can use this model to resolve the RNA 3D structure prediction problem. So this problem is similar to the protein structure prediction. Given the sequence, we predict the coordinates of each atom within the sequence, and then compare it against the resolved structure, right? So we built out a model called the RoFold Plus, okay? So the model itself is similar to AlphaFold, but there's one very different, significant difference. That is, we put the RNA foundation model into this model so that we can augment more information from the huge amount of database and retrieve more homologs. We tried to remove RNA-FM. We find that the performance will be reduced significantly. One reason is that we don't have enough data. And also, without this kind of RNA foundation model, it's relatively more difficult to try to retrieve the core evolution information. So this entire model is similar to AlphaFold. I won't introduce details here. So let's see the results. So we compare the RoFold Plus against AlphaFold 3. So the left figure is in fact from the AlphaFold 3 paper. They compared against our previous version called AIxBIO. So it's on the Cas15 RNA targets. So you can say part of the RNA AlphaFold 3 performance better. Some of the RNA, the performance of AIxBIO is better. And later on, we did some upgrades to our model. Now it's called the RoFold Plus. And on our recent Nature Methods paper, we compared against the different methods. So I would say the performance compared against the AlphaFold 3 is better. But honestly, the problem of RNA structure prediction hasn't been resolved. So you know, for the protein structure prediction, the usual MSD is around 3 to 4 Armstrong. But for the RNA structure prediction, you can see here roughly is 7 to 8 Armstrong. So there's still a huge performance gap between different fields. Okay, more data and more models should be developed to try to resolve the RNA structure prediction problem. Okay. So there are some more results here. We try to check the performance across different RNA type and different RNA family. So the one very interesting point here, RoFold Plus can correct some of the artifacts in the database, which are illustrated in the most left figure here. So when we try to resolve the RNA structure, because sometimes the RNA structure is not very stable, so researchers will try to form a dimer. That is, put 2 monomer RNA into a dimer so that to stabilize the structure and can resolve it using cryo-EM or X-ray crystallography. Then when we try to put the resolved structure into the database, you know, it's no longer a monomer, it's a dimer. And we still cut them into a monomer and then put it into a database. Then some part over there, they should form into a helix. But because we form into a dimer, so those parts will dehelix that. But this is actually not the natural state. So we found that our model can correct this kind of artifacts from database, which is very interesting. Okay. But of course, structure prediction is not the end of goal. Our final goal is to do some RNA drug discovery and RNA design. So it's a kind of the inverse problem against the RNA structure prediction. So given the structure of the target RNA, we want to design a sequence which can eventually fold into the structure. With that, we can have lots of applications like redesign the ribosome, ribosome optimization, optimal design, maybe ASO design, and so on and so forth. So back to last year, we developed 2 models to try to resolve this problem. The first model is called the Ribo diffusion. I would consider this as a prototype because we didn't perform the wet lab experiments to validate the performance. But instead, we performed some in silico validation. So there are 3 requirements for this task. First of all, we want to design RNA sequence to have high diversity against the native RNA. If the designed RNA sequence is very similar to the native RNA, then what's the meaning of redesign, right? So this is the first requirement. The second requirement is that given the designed RNA structure, we can design the RNA sequence that can eventually fold into the structure. That is, we satisfy our initial requirement. Okay? And ideally, the last requirement is that the designed RNA sequence can have better properties than the native sequence. That is the final optimization of the RNA. Okay? So here we show some in silico results of ribose diffusion, but we didn't perform the experiments. For the second model, we call the row design. So this is the inverse model of row fold. And for this model, given the structure, we can generate lots of sequences that can potentially fold into the structure. And for this model, we actually did some well-lab experimental validation. So we use the fluorescence Mongo aptamer as an example. So we designed 2 rounds of sequences. We asked the model to generate sequence with less than 50% sequence similarity. And for the first round, we did the well-lab experiment validation for 18 of them. And 4 out of them can be positive with the fluorescence activity. And we use this first round of data as additional training data to further fine-tune our model. And we did the second round of validation. We find that this round, the success rate is very high. Roughly 15 out of 20 can be positive. And very interestingly, 10 design aptamers can show higher fluorescence than the native Mongo 1 aptamer. Okay? And we also performed some functional assay to show those positive aptamers have experimentally validated the design RG4 structure, which is essentially the structure we put into the model. Okay? So in terms of speed, we essentially reduce the entire design pipeline from several months to 4 weeks. Okay? So this project is a very quick project. We essentially finished the project within 3 months. But of course, it takes 1 year to get the paper published. Right. Okay, so that's essentially the summary and a very brief introduction of the representative projects from our group. I do believe AR will be very helpful, especially the recent progress of AR will be very helpful to promote the research in the bioinformatics field, not only in protein, but also in RNA. Okay? So I'd like to thank all the collaborators. So those projects wouldn't be a success without my collaborators from different universities and different institutes. I also like to thank the founder from Hong Kong. Okay. Also thank my group members. So those projects are done by my PhD students, not by me. Okay. Thank you very much for your attention. Okay.
Okay, thank you very much for the very interesting talk about RNA modeling.
Yeah.
And I think we have about maybe 2 minutes to remain. So any question from audience?
Sure. Do you have any question? Okay, one question, okay. No question? If no question, we can probably discuss during coffee break. Oh, one question? Okay. Sure.
Hi, great talk. I think it's very clear that language models is very important for protein, not only for protein, but also for RNA. The question that I had was that a lot of these models, language models, use existing architectures that are developed for other purposes, not necessarily biology.
Yeah.
Right? And it seems like many people are developing their own RNA language model and own protein language model. I think like every single day there's a new protein language model. What is your perspective on like what's like the next step and what's like the bottleneck in terms of coming up with more informative language models for protein or RNA?
Okay, thank you. This is a very good question, and also this is something I've been thinking about. So I think in terms of a model, in terms of the model capacity, those existing models from the NLP field are already very helpful. So for those models, we essentially borrow their model architecture and borrow their model capacity. But in terms of the model training, what we need is the data, high quality data. I would say the next very important thing is to try to collect not just the protein sequence data, but try to identify the data interacting with protein. Those kind of affiliate data will be also very helpful. And next thing would be try to identify the real killing application, right? I would say those models, if you just consider a general model, then they're not useful. Only with the right application, with the killing application, those models will be helpful. Yeah.
Okay, thanks.
Yeah, thank you.
Okay, thank you again, Professor Yu Lee. I'm going to introduce the third speaker today. Speaker is Professor Do-Hun Lee. So please welcome Professor Do-Hun Lee. Sure. So I'm going to introduce Professor Do-Hun Lee briefly. He got his bachelor degree, master degree, and doctoral degree in computer science at KAIST. After this, he had so many professional experience. I'm going to have a list of a couple of experience, which is he's a former president of Korean Society for Bioinformatics, and also he's a former member of a Presidential Advisory Council on Science and Technology. And now she is a current member of the board member of National Research Council of Science and Technology. Also, he published so many papers which I can list out today, but including the cancer cell, which is these papers. So today he's gonna talk about context-oriented directed association networks for drug discovery. Please welcome Do-Hun Lee again.
Okay. Thank you for a kind introduction. I'm really happy to see Peja and the other close friend here. Actually, I should have come earlier this morning, but I had another meeting in another place of Seoul. So anyway, the topic is context-oriented directed association for drug discovery, but I changed it a little bit like this. Context-oriented directed association and biodata ecosystem. Actually, those two topics are a little different, but tightly coupled each other. So I'll explain why they are. So outline is multi-compound, multi-target strategy as an alternative strategy for treat very difficult disease. And then I will introduce our technology called CODA. Actually, in Korea, there are two CODAs. I will explain it later. It's an interesting story. And lastly, I will discuss with you about how we can share, exchange bio-research data in a voluntary way. So this is an interesting slide. Let us see the situation. The drug, medicinal agent, therapeutic agent, anything is an external stimulus to the homeostatic human body, right? So they have some functional efficacy as well as unintended side effect together. Because they are aliens to the human body, so we cannot expect all the effects are positive. The positive and negative together. This is quite. But so, if we want to get some efficacy, there's no, okay, it's better, sorry. Pointer does not work here. So, okay. Let's look at the left-hand side, single pharmacology diagrams. So if we want to get some efficacy, we need to increase the dose, right? But the problem is, as the dose is increased, the side effect threshold can be arrived, right? So to get the efficacy, we need to prepare the side effect. So that's the problem. If we use the therapeutic agent, the two drugs, then we can a little find detour ways. So in order to increase efficacy, we apply half dose of A drug and half dose of B drug. And they work together to get efficacy to get over the clinical threshold. But if we can assume that side effect of drug A and side effect of drug B are different, and they are still in their lower than threshold level. So we can avoid the side effect. That's the idea. But I found that the well-known Albert Barabasi also has the same idea as me. So his paper, he observed that if two drugs work together, then in much more cases, they control different parts of human physiology. So that is the B picture. I cannot use the pointer. Anyway. The A picture is overlapped function. So if two drugs act to the same point, then we cannot expect their synergistic effect. This is an empirical study. So maybe the left-hand side, conceptual ideation, and right-hand side, the empirical statistics are consistent. So I have put many effort to get some polypharmacology drugs, especially I collaborate with the Korean the natural product pharmacology company, pharma, called Amthera. And our agent called MT101 is a Parkinson drug candidate. And not only alpha-synuclein aggregation, that is the main target of most of the Parkinson's disease drugs, but also the neuroinflammation and mitochondrial dysfunction and synaptic disruption can be controlled by this mixed agent. So actually it is in the clinical trial stage 2 in the United States after passing the FDA IND approval. So, but the problem, the technical challenge is the single drug sufficiently difficult to develop. If we consider two drugs, the complexity becomes much more severe. Why? All possible cases we need to examine becomes increased exponentially, not linearly. So cell-based experiment or even animal-based experiment is impossible. That's why the computational model of human physiology is necessary. It's automatic. So I have developed some particular virtual human system where all kinds of molecular, cellular, and symptom-level interactions can be represented in terms of software code so that we can examine the effect of drugs in silico. I named this system like this, CODA, Context-Oriented Directed Association, because in biology, in many cases, it is very, very difficult to claim causality. In philosophical viewpoint, it is impossible. So we like to say association. A phenomena is associated with B phenomena. A drug is associated with B. That is actually honest claim. But we can come one step more, directed. In that case, A influence B associated, but A is some candidate causality of B. So this is a very humble representation of causality, directed association. But we also know that many directed associations depend on biological context. So this drug is good for me, but this drug is not good for my friend. So this is because the context is different. So context-oriented directed association may be one of the possible elementary knowledge unit we need to represent and accumulate it to simulate human physiology. That's why I named the system as like this. But one of my colleagues in the other Ministry of Health Science in Korea, they developed human clinical sequence archival system, very important system, and they named it as CODA without noticing me. So we did not discuss each other. So after like 3 or 4 years later, we found that we had the same name. So he told me that you should give the name to me. But I told him that you are from government department, so your system will go forever. But this is a very small the academic system, so just give us some patience. But the practical reason is, under this name, the patent and papers are already published, so I cannot ignore the name. So anyway, that is the history of why we Korea have two CODAs. I like him anyway. I don't hate him. This is the 4 years already, or not 4 years ago, Nobel Prize for Economics. What they awarded is, in social science and economics, it is very important to uncover directed association between some policy and the effect. But the problem is, it is impossible to conduct real experiment, not only biomedical area, but also the society area. In many areas, it is impossible. So when they design some social policy, they want to predict the outcome of the suggested policies in advance. So they utilized the past data. So by analyzing past data, they have suggested many indexes and the methodologies, and they found that the data-driven causality inference can be reliable in social policy design and optimization. So that is their core contribution deserving the Nobel Prize. So very similar to us. We would like to use the data to find causality, directed association between the biological phenomena. So this is a CODA system, and CODA system is actually just a computer program. It collects information from well-known databases, including TAG, drug bank, and so on, and it also parses the biomedical literature in PubMed repository, and it also collects information from our collaborating labs. And by integrating all the information, they can predict systemic function of some medical agent and systemic pathways of molecular and cellular interactions of the therapeutic agent. So the knowledge unit looks like this. Very simple. A activate B in context C. But for A, they can molecules or drugs or symptoms or cell behaviors, whatever. And the context can include the species, disease, and symptoms, and the other biological context. So this is a language to represent that basic knowledge, and all the information are coded based on internationally recognizable code system. And now it arrives at its version 3.0. So still we are going to on the way to version 4.0. I think that I have to do this research forever. This is a 3.0 level and screenshot. So in order to use this system, we need to select what network we want to utilize, diabetes network or cancer network or the infectious disease network, whatever. And the network has been, once the network is chosen, then you can apply. What if he takes or she takes some drugs or some herbs or some combinations of compounds, whatever, then what is the predicted effect or phenotype change? Then system will answer like this, the phenotype list. And second function is path exploration. If we know that particular therapeutic agent can increase some blood pressure or something, then what is the intermediate path? Then the CODA system predict that agent binds to particular protein, and that protein modulates some particular cellular function, and that cellular function controls some organ level functions, and it connected to the blood pressure and something like that. So that is the. So we have applied that technology to various applications, including drug effect prediction and drug repositioning and drug interaction and endogenous hormone and drug interactions, and even clinical trial design. Actually, that application, it was not my intention, but my collaborator found it. She was developing some particular natural product to cure woman. I cannot remember. After a woman becomes older, the hormone level changes and menopause. Thank you very much. Yeah, that is very critical to our colleague, woman scientist, not only scientists, but also or woman citizens, and menopause. So she was developing that one, and she found very effective ingredient from natural herbs. But she tried to design clinical trials, but she had trouble. It was hard to decide what endpoint should be included and what eligibility criteria should be included in the clinical trials. So she gave the information to us, and we analyzed the effect, and we found that it can control what pathways, and it can affect what phenotypes significantly. So after reading that report, their team designed the clinical trials, and they made a success. So I think that it already goes to the commercial market because it was not a drug, it was a functional food ingredient. So as I told you that the pharma, I'm sorry about the Korean characters in the slide, but this is a Korean one of the #1 newspapers, and they reported that our candidate drug is in the FDA clinical trial. So this is not the final slide. Actually, I am in the half of the total talk. It is not my own work. Actually, this interesting, wonderful technology has been developed thanks to all the collaborators. Not only these collaborators, but also worldwide colleagues, because they have provided information, data, through public databases, through publications, through the personal communication data files. So actually the technology like CODA is just one example which can be developed based on bio research data sharing and exchange. So I'm going into that topic seriously. I would like to claim that we need to prepare and we need to develop bio research data sharing platform and ecosystem and regulations and the culture. Why? Actually, data sharing is the way of science itself. Do you agree? Because we have, our ancestors and us and our descendants will share data through publication. Publication is the primary media of scientific discussions and science development. But the problem is only selected positive data that support the main claim are published. Do you agree? So it is the limit of current culture. But if we can smartly and fairly and effectively share bio-research data, then we can recreate data value. For example, the patient genome data, it has been collected in the hospitals for patient diagnosis and treatment. But by reusing that data, we can discover disease subtypes and disease diagnosis, the completely different technology which can help the patient, right? So it was not the original intention, but the data value is recreated. And new drugs and new indications can be discovered by completely different drug clinical trial data, if they can be shared. So it will open very rich area of value recreation. And the other reason, increasing research performance and usability. When I discuss with the experimental scientists, many of them like to say that this experiment works in lab A, but it does not work in lab B. Actually, that is true. I think that that is the fundamental difference of biology to physical science. Because physical science, they are dealing with non-living things. So their state is the same. So they can be reproducible. But biology, we are dealing with living things. So what is the meaning of living things? It is changing forever, continuously. So it means that reproduction is not an easy thing, completely different viewpoint. So, but in order to develop drugs and in order to make some useful tools, we need to find reproducible part of the biological phenomena. So in order to do that, we need to share the data. And lastly, now the Dr. Yu Lee and Professor Gang also mentioned how artificial intelligence can help us or maybe lead us. And so we need to collaborate with them. So in order to do that, we need negative data as well, not only positive data to work with artificial intelligence. That's why the bio research data sharing is necessary. Traditional way is the academic journals, they also provide to some extent the way of exchanging data. The long time ago, the hard copy-based system, because the limit of the space, it is very limited. But these days, most of the online academic journals, they are published online, so there is no limit of the space. Not really. These days, we are dealing with sequence, then it can knock the limit of the hard disk space as well. But anyway, in online publication. And there are many data-centric journals recently developed, including NAR is a really traditional classical data-centric journal, and GigaScience and Nature Scientific Data, and recently last year, the IEEE, they also started Data Descriptions Journal, which is aiming at data-centric journals. And database services itself, including the GenBank, ENA, and DDBJ, the INSCD, and the PDB, CAG, and drug banks as well. Many traditional media exist, but still, and one more, the NIH started this initiative from 2022. As you might be familiar with, the Harvard Dataverse and Dryad and Figshare, OSF, and those very wonderful, great platforms developed to collect not only biological data, but also scientific data and share it among the community. But the NIH may build a federation of those 7 platforms to curate them. So each platform works autonomously, but they are federated. How to federate them? The code mapping and the data format protocols are decided and harmonized. So this year, the Mendeley Data and Zenodo, those 2 platforms are having the chairman position. So that is not important. They change the leadership every year. In Korea, we also developed Korean National Biodata Station several years ago, and we really put our best effort to collect the human genome, not only human genome, but also the chemical structures and chemical assay data and toxicology data and many data. So, but it is not easy to collect data for sharing. This is a picture from Nature 16 years ago, and the University of Rochester started to collect data and share, but as you can see, the data archive is empty because data producers, they are really hesitating to data to the shareable platform. But this is a very interesting observation recently found. It's not from by myself. It is from Professor Kwon Seok-bum in Seungkyungan University, one of my colleagues. He found that by analyzing the NIH, the US publication data, if particular scientists disclose the data to the community, what happens? The citation becomes much higher than the others, as you can see in the picture. Right after the year 2010, he disclosed, or she disclosed the data, and relatively, the citation becomes dramatically higher than non-disclosed counterpart. Okay, so it is one reason why we have to. It is benefit to data producers to share data, and the other thing is actually this is actual worry of many data scientists. If I disclose my data, my competitor can get the data quickly and get the better research outcome than me. So it is not the true also in statistical observation. In many cases, data producer, if somebody get the data from that data producer, then the academic hypothesis and goals of these people, the data taking the scientist and data donor, might be many cases different, not the same goal. It is quite, you can think about that. If you take the other's data, then you know that he already produced the data and analyzed the data, and he's doing research now, so he's going that way. So now I just got the data. So I'm very behind of his progress. So if I pursue the same objective, it is obvious that it is not easy to go further than his steps. So that is his observation. So by taking this data fact-based observation, we need to change the community like this. Competition and cooperation should be merged into co-petition. Co-petition is a new word. You cannot find this word in the Oxford Dictionary because this word is coined by the NIH Gray Initiative. Dr. Susan, his family name is too long. I'm sorry about that. And Susan coined it. Yeah, Gregory, yeah. And it is a very important concept we need to share and expand to our community. And obviously, we need to prepare the motivations for data sharing scientists. So some people say that we need to prepare some monetary reward or something like that, but I think that is secondary. The primary is we need to provide academic credit to data sharing scientists. So we are investigating how we can materialize those motivations. And lastly, we need to build the data curation communities as well. The NIH already built it and operated, and we need to prepare that one. And we need to recognize the value of a secondary data set as well. And the last one, the centralized single data repository is impossible to our community. We need to respect each data repositories, not only Ministry of Science and Technology, but also Ministry of Health, but also Ministry of Commerce, and they can have their own authority and platforms. And we need to find a way how we build a federation, effective federation of those platforms to leverage the data value. Okay, this was my last slide. Exactly 30 minutes. Thank you very much.
Thank you very much, Professor Tu-Han Lee. And we have 1 minute left, so I can ask maybe one question from audience. If not, please thank again Professor Doan Lee. Please thanks again. Okay, so we're closing our session today. So we're gonna have a coffee break until 3 p.m. So please come back at 3 p.m. Thank you very much.
Thank you to our speakers and Professor Liu. As he explained, we have another coffee break now. Please enjoy the refreshments and return by 3 o'clock. Thank you.
Thank you very much. Oh.
Should I send to you my slides?
Oh.
By email?
We will soon start our next session, so please be prepared. Welcome back, everyone. Now we'll start the next session. Next, we have a special policy talk and chaired by Professor Sang-ho Kim.
Hello again. So before moving on to the next scientific session, we're going to have a short 15-minute talk for Korean policy to stimulate the AI bio. So this policy talk will be made by Mr. Hyuk-mo Nam. Mr. Hyuk-mo Nam is Director, Office of Advanced Biotechnology Division in Ministry of Science and ICT. And this talk will be about the Korean strategy. So it will be, the presentation will be in Korean. I'm sorry to say that, but I heard that all the slides are in English, so I think you can at least get some sense of that. So please welcome Mr. Nam.
제가 오늘 이 자리에서 정책을 발표해 주십시오라는 요청을 받았을 때 맨 처음에 여쭤봤던 거가 한국말로 해도 되냐라는 거였습니다. 오늘 이 한국말을 잘 이해하지 못하시는 분들도 계신데 그냥 한국말로 진행을 하게 돼서 송구하게 생각을 하고, 우리 옆에 계신 남준우 단장님께서 열심히 통역을 해주시겠다고 하셨기 때문에 믿고 한국말로 하도록 하겠습니다. 제가 이걸 준비하면서 자료를 좀 찾아보니까 구글 마인드 창업자인 허사비스 그분이 하신 생명체는 복잡하고 역동적인 정보 처리 시스템으로 수학이 물리학을 설명하는 언어이듯이 AI는 생명 현상을 이해하는 데 가장 적합한 도구다라는 표현을 찾았습니다. 아마 오늘 논의를 하고 있는 많은 주제들이 기저에 깔려 있는 말이 아닐까 생각을 합니다. 글로벌 빅테크 기업이 연이어 바이오 분야에 진출하면서 AI 바이오 플랫폼 주도권을 둘러싼 경쟁이 본격화되고 있는 바이오 대전환 시대에 AI 바이오 정책이 어떻게 나아가야 되는가에 대한 저희의 고민을 좀 말씀을 드리도록 하겠습니다. 목차는 패러다임의 진화, 그리고 그간의 디지털 바이오 육성을 위한 노력들, 그리고 향후 계획으로서 저희가 준비하고 있는 것들을 간략하게 말씀드리도록 하겠습니다. 먼저 바이오 패러다임의 진화입니다. 이 슬라이드는 다들 아시는 내용일 것 같아서 간략하게 지나가고, 근대 생명과학의 시작은 생명 현상에 대한 이해, 과학적인 근거를 통한 기초를 확립하고 20세기에는 그 원리를 발견하고 응용함으로써 새로운 치료법을 개발하는 그런 발전의 과정이었다고 할 수 있습니다. 바이오 연구는 복잡한 생명 시스템을 탐구 대상으로 삼고 있기 때문에 전통적인 실험 방식은 장기, 고비용, 불확실성의 한계에 직면할 수밖에 없었습니다. 21세기를 흔히 첨단 바이오 시대라고 이야기를 하고 있습니다. 이제 이해나 발견에서 새로운 발명의 시대로 가게 됐고, 보다 더 효율적인 그리고 효율적이고 저비용으로 할 수 있는 다양한 기술들의 진화가 이루어졌습니다. 특히 AI의 경우에는 단순한 연구 보조 도구로서의 기능을 넘어서서 R&D와 바이오 산업 전 영역에서 패러다임의 혁신을 선도를 하고 있습니다. 2018년인가요? 구글 딥마인드의 알파폴드가 수년이 걸리던 단백질 3차원 구조 예측에 소요되던 시간을 단 며칠로 해결을 함으로써 생명공학계에 큰 충격을 주었습니다. 이후에 AI가 실제 생물학적 기능을 지닌 단백질 분자를 설계하는 수준에 도달을 했고, 이제 그걸 넘어서서 AI가 연구 목표를 설정하고 가설을 생성하며 실험 설계 등을 종합적으로 지원할 수 있는 수준으로 발전을 해왔습니다. 이 슬라이드는 그간 AI 바이오 혁신의 연대기라고 생각하는 주요한 사건들을 정리를 해본 자료입니다. 실제 사람의 생체 분자들을 가지고 연구를 진행해 오던 거에서 2010년대 초에 가면 딥러닝의 혁신이 일어나게 되고, 2010년대 후반에는 AI 기반으로 한 단백질 구조 예측 모델이 나타나게 됩니다. 이후 현대에 이르게 되게 되면 단백질 구조 예측뿐만 아니라 설계, 그리고 생체 분자 구조 설계로의 확장이 이루어지고 있고, 아래에 보시는 처럼 국내 AI 바이오의 경우에도 세계보다는 조금 늦은 감이 있긴 하지만 꾸준히 발전을 이룩하고 있습니다. 현재 AI 바이오 시장에 대해서 먼저 간략하게 말씀을 드리고자 합니다. 글로벌 AI 바이오 시장은 24년 32.3억 달러에서 29년 77.5억 달러 수준으로 연평균 19.1% 증가할 것으로 전망을 하고 있습니다. 학술적인 측면에서 미국이 최상위 수준을 확보를 하고 있고 한국이나 중국과 같은 아시아권 국가는 기술 개발 활동이 활발하게 이루어지고 있습니다. 활용 분야 측면에서는 신약 개발, 단백질 구조 예측, 설계, 유전자 치료 등 바이오 분야 전 영역에서 전례가 없는 혁신이 이루어지고 있습니다. 이러한 AI 바이오 주도권을 선점하기 위해서 주요국들은 고품질의 바이오 데이터 플랫폼을 구축하고 바이오 연구에 필요한 인프라 확충 등에 대한 지원을 강화하고 있습니다. 왼쪽 편 아래쪽에 보시게 되면 바이오 데이터 구축과 관련해서 잘 아시는 것처럼 미국은 올오버스 프로젝트를 통해서 공공주도로 100만 명의 보건의료 데이터 수집을 하고 있고요. NIH에서 브리치2 AI 프로그램을 통해서 바이오 의료 분야의 AI 학습에 최적화된 고품질 데이터를 생산, 활용하기 위한 프로그램을 진행을 하고 있습니다. EU의 경우에도 유럽 게놈 데이터 인프라 프로젝트 를 22년부터 추진함으로써 유전체 및 임상 데이터에 대한 접근성을 제고하기 위한 노력을 추진을 하고 있습니다. 오른쪽 부분은 인프라에 대한 부분입니다. 미국의 경우에는 바이오 제조 생태계를 구축하기 위해서 21년부터 바이오 메이드 프로젝트를 추진을 하고 있고 유럽의 경우에는 바이오 제조 혁신을 위한 규제 간소화 정책을 24년도에 발표함으로써 AI 기술이 바이오 제조 혁신의 핵심 요소로 자리 잡을 수 있도록 지원하겠다고 공언하고 있습니다. 일본의 경우에는 바이오 제조 혁신 전략을 통해서 바이오 제조 생태계 조성을 위한 민간 합동 투자를 확대하겠다고 발표를 한 바가 있습니다. 이런 상황에서 우리나라의 상황을 조금 보게 되면 가장 왼쪽 편이 정부 R&D 투자 규모입니다. 긴 막대기로 되어 있는 게 정부 바이오 분야 R&D 전체 투자고요. 이거는 조사 분석 평가 결과로 나온 거여서 5조가 조금 넘는 바이오 R&D를 투자를 하고 있고 파란색 부분이 AI 바이오 부분입니다. 근데 요거는 저희가 그 NTIS 과제 중에서 인공지능이라는 키워드를 가지고 검색을 한 거기 때문에 실제보다는 좀 많이 적게 지금 산출되어 있는 겁니다. 24년도 기준으로 306억 원인데 그 키워드로 검색한 거라는 걸 좀 양해를 해주시길 바랍니다. 이게 조금 더 늘어난다고 하더라도 실제 전체 바이오 R&D 투자 대비해서는 비중이 굉장히 미미한 그런 상황입니다. 다음으로 생태계 쪽을 살펴보게 되면 저희가 지난 24년 9월 달에 전문가분들을 대상으로 설문조사를 했습니다. 혹시 연구를 하시는 데 있어서 AI를 활용하십니까? 라는 질문에 52%의 전문가분들이 AI를 활용을 하고 계시다고 답변을 해주셨고요. 지금 현재 AI를 활용하고 있지 않더라도 향후에 AI를 꼭 사용을 하고 싶다라고 밝히신 분이 지금 활용하고 있지 않으신 분들의 96%에 달했습니다. 그래서 연구적인 측면으로 보게 되면 평균 피인용도가 세계 3위권에 있을 정도로 기술력에 있어서는 상당한 역량을 가지고 있다고 판단이 됩니다. 산업 부분에 있어서는 독자적인 AI 신약 개발 플랫폼 구축을 추진하고 있지만 아직 초기 단계에 있어서 뚜렷한 성과는 미미한 상황입니다. 앞서 말씀드린 정부 R&D 투자액이 적다는 것뿐만 아니라 실제 AI가 바이오에 활용이 되기 위해서는 컴퓨팅 자원이 필수적입니다. 이 컴퓨팅 자원이 너무 부족한 상황이어서 해외의 의존도가 높은 상황이고 고품질 다유형의 데이터가 마련이 되어져야 되는데 저희가 그러한 데이터들이 부족합니다. 22년도에 구축하기 시작한 국가 바이오 데이터 스테이션은 해외 대비해서 늦은 출발이고 그래서 아직 초기 단계에 있습니다. 이러한 상황을 고려했을 때 선도 국가로의 도약을 위한 혁신적인 전략이 필요한 상황이었습니다. 그래서 그간의 디지털 바이오 혹은 AI 바이오 육성을 위한 노력들을 간략하게 말씀을 드리고자 합니다. 지난 22년부터 시작을 해서 정부에서는 AI 바이오 혹은 디지털 바이오를 육성하기 위한 다양한 전략과 정책들을 발표를 했습니다. 22년도에 디지털 바이오 혁신 전략, 그리고 23년도에 제4차 생명공학 육성 기본계획, 24년도에 첨단 바이오 이니셔티브에서 공이 AI 바이오의 중요성 그리고 이를 활성화하기 위한 다양한 전략들을 포함을 하고 있습니다. 먼저 22년도에 발표한 디지털 바이오 혁신 전략에 대해서 간략하게 말씀을 드리도록 하겠습니다. 가장 왼쪽 편에 보이는 것이 5대 인프라입니다. 휴먼 디지털 트윈, 소재 플랫폼, DNA 바코드 화합물 은행, 그리고 국가 바이오 데이터 스테이션, 그리고 바이오 파운드리, 이 5대 인프라, 그리고 오른쪽 편에 보시게 되면 첨단 뇌과학이나 디지털 치료제, 전자약, 오가노이드, AI 신약 플랫폼 등 12대 핵심 기술 확보를 목표로 삼았습니다. 그래서 이 인프라 구축과 기술 확보를 위해서 저희가 R&D 투자를 확대하고자 하는 내용이 주 내용이라고 생각하시면 될 것 같습니다. 조금 더 디지털 바이오 혁신 전략에 대해서 구체적으로 말씀을 드리게 되면 왼편에 있는 것이 4대 추진 전략입니다. 디지털 바이오의 신기술 신산업을 창출하고 기반 기술을 확보하고 데이터 기반의 바이오 연구로 확산, 그리고 이를 위한 생태계를 조성하는 전략입니다. 먼저 바이오 분야의 IT 기술을 접목을 해서 연구 패러다임을 전환을 하자라는 내용입니다. 그리고 바이오 기술로 석유화학 제품을 대체함으로써 제조 산업을 혁신하겠다. 그리고 새로운 기전과 접근 방식에 기반해서 그동안 극복하지 못했던 질병에 대응할 수 있는 역량을 강화하겠다라는 것이 주요 목표라고 생각하시면 될 것 같습니다. 이를 위해서 데이터 기반 유전자 세포 분석, 제어 기술을 고도화하고 연구 속도와 성공 확률을 향상을 하고 국가 바이오 데이터 공유 플랫폼을 완비하고 글로벌 수준의 데이터 관리 체계를 구축한다라는 내용들, 그리고 민관 협력과 국제 공조 확대, 그리고 실질적으로 AI 바이오를 할 수 있는 융합형 인력 양성이나 분야 간의 융합 연구 활성화에 대한 내용들을 포함을 하고 있습니다. 다음은 제4차 생명공학 육성 기본 계획입니다. 생명공학 육성 기본 계획의 주요한 4대 전략 중에서도 제일 왼쪽 편에 첫 번째로 삼았던 것이 디지털 융합을 통한 바이오 혁신 부분입니다. 이 부분은 간략하게 4대 전략이 나와 있는데 바이오 기술과 다른 기술을 접목을 해서 핵심 분야의 부가 가치화를 하겠다는 내용, 그리고 바이오 기술을 다른 분야에 접목을 해서 소재나 제품 개발 등 제조 산업을 혁신을 하겠다. 비슷하게 디지털 융합형 바이오 기반 기술 확보, 데이터 기반의 바이오 융합 연구 활성화를 하겠다라는 내용이 포함이 되어져 있습니다. 근데 뒤에 2번, 3번, 4번으로 나와 있는 글로벌 난제 해결이나 바이오 경제 성과 창출을 위한 스케일업 촉진, 그리고 바이오 융합 생태계 조성, 이 부분에도 AI 바이오는 다 공통적인 기반 기술로 생각이 돼서 저변에 다 깔려 있는 거라고 생각을 해주시면 될 것 같습니다. 다음으로 24년 4월 달에 마련이 된 첨단 바이오 이니셔티브입니다. 여기는 바이오 가치 사슬 강화라는 방향성을 가지고 있습니다. 혁신 기반 기술들이 고품질 데이터를 만나서 서비스 플랫폼에 얹혀지게 되면 국민들께서 체감할 수 있는 체감 가치를 구현을 하겠다 하는 게 기본적인 방향이고요. 여기에 디지털 바이오, 바이오 제조 혁신, 바이오 의료 혁신, 인류 공동의 난제 해결이라는 부분이 중점 추진 과제로 되어 있고요. 특히 디지털 바이오 육성에서는 바이오 데이터의 확보, 그리고 고도화, 활용, 그리고 AI 플랫폼 개발에 중점적으로 작성을 하였습니다. 그럼 이러한 전략에 기본해서 지금까지 디지털 바이오 육성을 위해서 해왔던 것들에 대해서 간략하게 말씀을 드리도록 하겠습니다. 제일 왼쪽 편에 있는 것은 지난 2022년도에 시작을 한 인공지능을 활용하여 신약의 후보 물질을 도출하고 인공지능 신약 개발 생태계를 조성을 한다는 그런 사업입니다. 오른쪽 편에 있는 것들은 지난해 24년도에 시작한 연합학습 기반 신약 개발 가속화 프로젝트입니다. 신약 개발에서 필수적인 ADME와 안전성 예측을 위해서 다양한 병원이나 연구기관, 기업들이 가진 데이터를 연합학습을 이용하여 학습시킨 모델을 개발하고 독점 데이터 공급 6만 건을 통해서 중앙집중 모델 대비 99%의 정확도를 갖는 모델을 개발하는 것을 목표로 추진을 하고 있습니다. 다음으로 기술 개발 외에 데이터나 인프라 구축 측면에서는 아래쪽의 그림에서 보시는 바와 같이 국가 바이오 데이터 스테이션에 대한 모습입니다. 왼쪽 편에 과기정통부, 해수부, 농진청, 환경부, 질병청 등이 참여를 하게 됐고요. 여기서 나오는 성과들을 같이 연계해서 등록을 하고 있습니다. 그리고 유전체, 단백체, 대사체, 화합물, 이미지 영상 등 5개 분야의 데이터 고품질화도 추진을 하고 있습니다. 여기에 추가적으로 25년, 26년도에 하고자 하는 것은 고성능 분석 활용 시스템을 추가를 하고 범부처 데이터 맵을 구축을 함으로써 데이터 활용에 대한 편의성을 제고를 하려고 하고 있습니다. 그리고 현재 공공 데이터 위주로 되고 있는 것들을 민간 데이터까지 확대를 할 계획입니다. 오른쪽 편에 있는 것은 25년 올해부터 새롭게 시작하는 바이오 파운드리 인프라 구축 사업입니다. 이 사업 외에 소프트웨어를 개발하는 사업, 그리고 기기를 국산화하는 사업도 추가적으로 병행해서 추진을 하고자 합니다. 다음으로는 법 제도적인 기반을 마련을 했습니다. 생명공학 육성법 개정을 통해서 AI, 디지털 등 다른 분야 기술과 생명공학 기술 간의 융복합 연구를 촉진하는 근거를 명시를 했습니다. 그리고 오른쪽 편에 보시는 것처럼 세계 최초의 합성생물학 육성법을 제정을 해서 합성생물학 연구 기반 구축 및 환경 조성, 안전한 연구 환경 조성, 생태계 조성 및 사회적 수용성 확대에 대한 정부의 노력을 명시를 했었습니다. 다음은 저희가 준비를 하고 있는 계획에 대해서 간략하게 말씀을 드리도록 하겠습니다. 올 상반기 중에 아마 빠르면 이달 말 정도에 발표를 하게 될 것 같은데요. AI 바이오 확산 전략입니다. 디지털 바이오라는 게 조금 범위가 좀 모호하기도 하고 많은 영역을 커버를 해야 하다 보니까 이런 부분 중에서 조금 더 AI 바이오에 집중을 해서 AI 바이오를 어떻게 확산을 할 수 있을까에 대한 부분으로 확산 전략을 마련을 하고자 합니다. 이 부분은 먼저 핵심 기술에 있어서는 AI 바이오 시스템, AI 신약, AI 정밀의료, AI 그린 바이오에 대한 핵심 기술 개발과 신규 사업들을 추진할 내용들을 담았습니다. 그리고 인프라 부분에 있어서는 자동화 실험실을 비롯해서 연구, 제조 자동화를 위한 필요한 인프라와 고성능 컴퓨팅 인프라 확충, 그리고 AI 바이오 안전성 강화에 대한 내용을 담았습니다. 다음으로 데이터 부분에 있어서는 이게 가장 중요하고 가장 안 되고 있는 것 중에 하나라고 생각을 하고요. 바이오 데이터의 구축, 연계, 활용을 위한 전주기 지원 강화뿐만 아니라 민관 데이터 연계, 활용 환경을 개선하는 내용들을 중점적으로 담았습니다. 그리고 인재 양성 부분에서는 융합 인재 양성뿐만 아니라 해외에 있는 우수 인재 유치하는 내용, 그리고 실제 우수한 연구자분, 전문가분들이 모여서 논의하고 새로운 아이디어를 창출하실 수 있도록 AI 바이오 혁신 거점을 구축하는 내용들을 담았습니다. 이 바이오 확산 전략 안에는 26년도에 신규로 저희가 해야 하는 것들, 그리고 하고자 하는 것들에 대한 내용을 좀 담았습니다. 아마 저희가 지속해서 전문가분들을 모시고 의견 수렴을 진행을 했는데 남은 기간 동안에도 여러 전문가분들께 관련된 내용을 좀 여쭙고 저희 이 계획을 조금 더 조금 더 실현 가능성이 있는 정책으로 만들기 위한 노력을 진행을 하도록 하겠습니다. 제가 어느 포럼에 갔을 때 축사를 한 내용 중에 변화의 한 가운데에 서서 그 변화를 몸으로 직접 느끼는 것은 정말 황홀한 경험인 거와 동시에 무거운 책임감을 느끼는 일이다라는 말씀을 드린 적이 있습니다. 지금 후대의 어느 시점에는 오늘 이 자리가, 그리고 지금 이 시점이 아마 변화의 가장 중요한 시점이라고 판단할 수도 있을 것 같고요. 그 변화의 중심에 오늘 여러 전문가분들이 함께 해주고 계시는 거라고 생각하고, 정부 관계자의 한 사람으로서 오늘 정책을 발표하면서 무거운 책임감을 가지고 여러 전문가분들이 연구하시는 데 있어서 조금의 도움이라도 드릴 수 있도록 노력하도록 하겠습니다. 감사합니다.
Thank you, Mr. Nam. I think we don't have time for question right now, but I hope that further discussions will continue in like dinner time. So the next scientific session will begin instantly, and Professor Sung Won-jung will be the chair of this session. Thank you.
Thank you, Director Nam and Professor Kim. Now we proceed to scientific session 2, and actually I am the chair of this session, so I move to chair's place. Okay. Our first speaker is Professor Martin Steiger from Seoul National University. Professor Steiger is well known for developing innovative bioinformatics tools, greatly impacting protein sequence analysis and structural biology research. Please welcome Professor Martin Steiger.
Okay. Thank you so much for the nice introduction and giving me the chance to talk here. I want to talk today about supercharged protein analysis in the era of AI. This is a title I use all the time, so people always think I talk about the same thing, but it actually changes. But I think it's a very general purpose title. So I'm going to talk about the methods that we develop and the ideas how to look at proteins in different ways. But before getting started, I want to introduce my group. So this is the group at the Seoul National University. And the mission of our lab is to accelerate biological science through fast and easy to use open source methods. So again, very broad. A lot of the methods come in the idea where you have a lot of like big biological data and just somehow extract knowledge from this biological data. Since I wanted to just give a brief introduction to biology, the central dogma. So you're probably all familiar with it. You have DNA, then you go to RNA, then you go proteins. These proteins are composed of these 20 amino acids, but they do have a structure, a three-dimensional structure. And this three-dimensional structure like implies the function, right? So these kind of like macro machineries can somehow do things by their distinct three-dimensional structure. And I think they're really, really exciting. So these protein structures and these functions that we have are quite exciting. There's a lot of like problems that we have, pressing problems in nature and that we have to solve. And I think these proteins can help. For example, you can use these proteins to tackle plastic pollutions by like breaking these plastics apart. You can use it for carbon fixation. So there's some bacteria that captures carbon and drag it down to the ocean. You can use it for medicine, so it's a repertoire of like medicines, of antibiotics and so on that are encoded in these proteins. Or climate change resilient crops, so the project that the Novo Nordisk Foundation started to fund is the one that I'm involved in, where we try to re-engineer old mutations that occurred before in hotter climates back into plants to make them resilient to climate change. And I think that all has to do a lot with the machinery and the proteins that are in these plants at the end. And so how do we tap into this potential of proteins? And one way of doing so is using metagenomics. So in metagenomics, we sequence environments, and then from these environments, we extract the genetic material, we put into a sequencing machine, and then we get reads out and contexts out, and they're encoding these proteins. And once you have them, you can then start questions about these environments. You can take these reads and you can search them against reference genomic databases, and then you can say, what is in that community? What organism is in that community? You can search against, with these reads against protein databases, and then you can figure out what do they do, like what kind of function do they encode, what proteins are there, and what are their potential. Or if you want to be really fancy, you can try to reconstruct metabolic pathways, like how are these organisms communicating with each other, and like what are they processing in full pathways. But a big problem that stays here is that many of these proteins remains unannotatable. So it means we cannot find them, we cannot match them back to anything in the databases. This can be bad. Up to 90% of the data sometimes cannot be used. So that means there might be these solutions, but we don't know what they are. So we need something. But before going into how to solve the problem of the annotation, I wanted to give an example of why I think these microbial proteins are really exciting. And one is like a collaborative work here between Wonju Song's and Myung-Sop Lee's lab, and we tried to find an enzyme for immunotherapy in huge protein databases. And so this is as much biochemistry as you get from me. So what you have is, here on the left is a cancer cell, On the right side, you have a T cell. And so these cancer cells somehow try to protect themselves by by blocking the T cells. And they do that with one mechanism, is this like alkyne ligand that they put out and they block the T cells. And we have a natural protein that helps us with this, with curinase, and that can break down this ligand into these two components. And in that study from Triplett et al., in 2018, they have actually found that there is a bacterial version of that protein, of that enzyme, that can do it much, much better. And if you give that to mice, you actually can increase the lifespan of the mice substantially. So we had a question in mind. The question is, can we do better than that? Can we find a better protein, like one that make the mouse even live longer with cancer? So here, you start off with the normal one. Here with the best one that found in the Triplett et al. Study is this PFK. You can shrink the cancer by 37% and increase the survivability of the mouse. So the question is, can we do better? Can we find something that shrinks it even further? And now my question normally comes, how would you do that? If somebody asks you the question, how would you do it? I'm not expecting answers, but just thinking about it would be good. So an obvious solution is to start off with that one protein that does well, right? So you take that one that is the best and you search in a database. You find things that look similar to the best one. Maybe there's a similar looking one that is actually even better. That's what we did. So you take that protein, we use a software that we have developed, MM6, and we searched against a big set of protein databases, and we find now many, many things that are homologous to this protein, that are looking somehow similar, have a similar amino acid composition. And what you end up with is a problem. You have 10,000 candidates. Now, I could go to Professor Song and say, hey, can you check all 10,000? And they would say, no, you're crazy. We can do 5. So we somehow have to break down that problem, right? We have to somehow prioritize that list in a better way. And there were a lot of talks about protein language models, and this is where we actually effectively will get to. You can use experimental values or experimental measures to somehow prioritize your enzymes. So in this case, we had about 159 data points that we could use. There were measurements for enzymes in previous studies where we know what the efficacy of these are. And so somehow we want to use these 159 data points. The problem is if you want to train now a big neural network, you cannot, right? You cannot train millions of parameters on 159 data points. So here's where really the power of language models come in. So language models are trained and unsupervised on big protein sets. And you get these embedding vectors, right? We have heard a lot today by Sun Kim in the morning, also Bonnie Berger, about these vectors. And these vectors are very powerful. So what you can do is you can turn a protein into a vector, and you can associate that vector to the experimental measure, and then you can train a small regressor model on these embeddings, like saying going from X to Y with like a random forest, for example. And if you do so, you get a Spearman correlation of 0.81. So it's like quite a good predictor, actually, just by a little bit of data you have. So we did that, we searched, we found our 10,000 candidates, we get these embeddings, we use our regressor model, and then we resorted the list of enzymes based on what the regressor thinks is the best. And you can see the top hit is now the hit that was previously at position 143. And all of the ones that we have tested, the top 3, they did all perform as well as the previous one, but the third one, the one I'm highlighting in red, which was at position 434, is actually doing substantially better. And for that one, we actually went into the animal experiments, and we could see that by giving this enzyme, you can shrink the cancer even further. So meaning that with very little data, you can do quite powerful things, right? You can imagine like a lab in a loop kind of thing where you generate a few data points, you make a regressor, and you find some, make new measurements, make a new regressor, and so on. And we have made that available for everybody, so you can just give a file with experimental measures and proteins, and we will, in the background, do a search for you, we will generate a regressor for you, and we will re-rank the hits for you. It's called seqrank, so you can just use that URL. But then now the big question, what if we don't find any homologous sequences, right? Like this is kind of like reliant on, we have to at least find something in the database, but if you do not, what will you do, right? This is how bioinformaticians look at similarities between proteins. So this is an alignment between two proteins. And normally at this point, I ask the question, are these two homologous proteins? Raise your hand if you think they are homologous. Okay, good. So there are some amino acid similarities between them, but just visually it's not super clear. Some things are similar, some things are dissimilar. But if I show you the respective three-dimensional structure and I would ask you the question, are these two objects similar? I think it becomes more obvious to you, right? They are actually similar. And the reason for that is that structure is just much more conserved and over much longer time. And so we want to use structure, right? Actually kind of structure for comparing proteins. For the majority of time we have used amino acids. But now, because of AlphaFold, and I think I don't have to show that plot, everybody knows AlphaFold does pretty well in predicting the structure, we do have access to a lot of predicted structures that are actually nearly close to the experimental accuracy levels. And yeah, the protein folding problem, right? How does AlphaFold work? Just a brief introduction. But before the protein folding problem, you start off with an amino acid sequence, right? If you put it into solvent, what it does is it goes down this energy funnel here, right? And at one point, it finds that native state. And that native state is where you have your three-dimensional structure, right? This is the happy point, like lying in a hammock in an island. That's where the protein is, like energetically, really happy. And this is kind of like what you want to do for structure prediction. And AlphaFold does that by taking a sequence, it builds a multiple sequence alignment. We'll talk in a little bit about what a multiple sequence alignment is. And it gives it then to a neural network that extracts from the multiple sequence alignment information, and it gives to a structural module that then turns it into three-dimensional coordinates, and it recycles that information of the structure back a few times to improve the prediction. So that's kind of like really like a rough sketch of how AlphaFold works. But so how does it do it? The MSA is effectively cheating. AlphaFold is cheating. It doesn't predict the structure from a sequence alone. If you just give it a natural sequence, it will not do very well in structure prediction, actually. It requires a multiple sequence alignment, and that multiple sequence alignment somehow constrains the problem for it. So you can imagine that the MSA is a pointer into the energy landscape, and the structural module is then a way to like refine the structure at the end, and you get the final structure out. So multiple sequence alignment, you start off with a protein sequence, you search against a database, and you find homologous sequences from different organisms. From that multiple sequence alignment, you can actually ask questions about your initial protein. So you can see this like a green point here. This is a point where you don't want to change the amino acid, like it's conserved over time. On the right, you can see this like yellow, red, green. That seems to be places where you can easily change the amino acid. And then you have this like blue, yellow, blue patterns that are like alternating. And they are these like co-evolving one. And these co-evolving ones are really critical at the end for the folding problem because they're often in physical contact. And these physical contacts helps actually to constrain the structural problem. So AlphaFold has a chance to predict that. So a lot of effort aside from the neural network, was also put into the information retrieval stage of AlphaFold, where you actually go through these big databases and you find the homologs. This is my part where I helped the DeepMind team for the AlphaFold system. I generated one of these big databases called BFD. So they searched with many tools against these big databases. One point was raised before. These searches are slow, and so these slow searches are causing actually problems if you really want to run AlphaFold on many proteins. So we have built a system where we took all of these databases and we combined them, and we used a different search tool, our MMCX2 alignment tool, which can find homologous faster than the previous tools that I mentioned in the previous slide. And we built a tool that's called CollabFold on it that has a web interface where you can use AlphaFold really simply just from your browser. This is the case AlphaFold 2. And we're building like different kinds of MSAs, one that you can use for monomers or one you can also use for multimers. And you can also use it locally on your Linux system. You can install just CollabFold locally, and you can, if you have a gaming GPU at home or so, you can actually use it for folding your own proteins at home now. So a bit about it. So if you go to collabfold.com, that's how it will look like, and you can put in a protein sequence, and what will happen is, if you push this run all button after a few minutes, a minute or so, you get like a protein structure back, you get prediction quality of it back, and you can figure out how the MSA looks like. CollabFold became really popular somehow, and actually by now, the back end, which we use for generating these multiple sequence alignments, are critical for many of tools that are used currently for modeling protein structures. One being Boltz1 or Chai1 or BioEmo. You might have heard of them in some of the recent articles. They all kind of use the MSAs that we are generating on our servers. Collab is not just in a Collab environment, like not just in the browser, but you can also run it locally on Linux and macOS. And I compare CollabFold a bit to like a medium successful K-pop video on YouTube. So it in the end has 56 million views, 56 million users now. So this is actually quite substantial, and it's hosted at Kobik. So I think I would say it's one of the biggest used resource in Korea for bioinformaticians. And I've mentioned AlphaFold is there, right? AlphaFold gives you the chance to somehow get these many predictions, and so they did have this AlphaFold database that grew to 214 million structure predictions covering nearly every protein in the UniProt. I mentioned nearly everyone. Actually, they excluded viruses. Viruses are not in. So we wanted to somehow complete the AlphaFold database. Therefore, we predicted viral structures. We took proteins from viruses, clustered at 30% sequence identity, about 350,000, and we predicted them with CollabFold. These cover about 3.2 million protein sequences in the UniProt. So it's really like a quite representative set of proteins for viruses. But we have a big problem. Many of these viral multiple sequence alignments contain less than 30 sequences, and that often causes troubles to AlphaFold. I mentioned AlphaFold doesn't do well on single sequences. If you don't have enough MSA information, you get quite a bad prediction for AlphaFold. So we ended up with many models that did not really fold well. But we had some work before, and where we have shown that you can actually do much better if you search through the whole SRA to build a multiple sequence alignment. So we have heard talks about data deposits, right? The SRA is one of the biggest ones. It's like petabases of sequencing reads that are just available publicly, right? Just hard to search through, hard to organize somehow. We need algorithms to go through them. So here we have used infrastructure in AWS to search through them and enrich the multiple sequence alignment through these petabases. And we did that for a CUSP-15. So we redid the CUSP-15 afterwards with all of the ones, and we just checked how much better would we have done if we would have searched through everything in CUSP. So initially, our structure prediction software would be ranked at position 11. And with the MSA depths increased, we would have been on position 3. We haven't done everything else. Everything is just effectively CollabFold with AlphaFold in the end, just running AlphaFold, but having a better MSA. And that actually would help you in that competition significantly, right? So we thought, why don't we do the same thing for all of these viral structures that had not enough coverage? So here you can see the fraction of predictions based on quality of it. So you see high is blue and very high is dark blue. First, we searched through Logan, which is an assembled version of the SRA. And with this, we increased, we got up about like 7 points or so. Then we did multiple recycles. We have seen this recycle mechanism of AlphaFold. We just did 12. And we then picked the highest PLDT out of the 3, and then you go up by more than 10 points in high quality structures. So these are tens of thousands of structures that have now really good quality. And you can explore these structures by going to bfvd.foldseek.com. You can search your protein structures in our database. You will then find a few similar ones. You can now select the one that you like, and then you can see the clustering and the structure that we have modeled, as well as the distribution in the database, and similar looking structures as well. And they should be now overlaid here to each other. Okay. So this search that you have actually seen is quite critical, this like structural search. I mentioned the problem of annotation, right? What if you don't find any homolog in the database? And this brings me to FoldSeek, which somehow is a method that is a fast searching method for protein structures, and a collaborative work with the Söding group in the Max Planck in Göttingen. So how can you be fast when you want to search structures? So structures are three-dimensional objects. So if you can compare structures in three-dimensional space, right? 3D against 3D. Unfortunately, that isn't very fast. So you can now reduce the problem to 2D. So you can compare, for example, the contact maps or something that describes the structure better and do that. Or you can go to 1D, just trying to describe the whole structure here as a string that is just similar to an amino acid string, but this is not encoding amino acids now, but instead we are encoding structural information. So just an alphabet that describes the structure, right? How would you do that? How would you get to a sequence that describes the structure? in biology, we had this for a long time. Most of the time, for example, the secondary structure was just encoded as 3 letters, right? Like an H, a C, or an E. And what we often did, we took the backbone torsion angles and encoded them into these letters. But somehow that isn't great for similarity search. The problem is if you encode these secondary structure elements, you would just end up as H, 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 right? And if you take that one and you search it in the database, it's everywhere. So you have effectively not gained any information. So we wanted to have an alphabet that somehow encodes more information. So what we did is we had a residue we want to encode here in red. The blue is the nearest neighbor in 3-dimensional space. And then we extract like vectors from them that describing the torsion angle between both of them and the distances, and so like a geometric descriptor. And we use a VQVE neural network to discretize it into 20 letters. And so then we have our 3DI alphabet. And that 3DI alphabet should contain more information, because now you actually describe parts of the structure, right? Like nearest neighborhoods. And to show that, to visualize what it does it do is, so you have here an alignment at sequence identity 16%, amino acid alignment. What you can now replace the amino acid information with the 3DI information, and if you do so, you end up at 54% similarity between them. And you can actually align that and see the similarity much easier. So 3DI really helps for this. And I've mentioned metagenomics, right? Like many of these open reading frames in metagenomes cannot be annotated. There was a study by Greg Lohr's group at Western University in Canada. They worked on wastewater phages. And these wastewater phages are hard to annotate. They've used a tool called BACTA, which use amino acids, HMMs, and whatever we have used before for annotating proteins. And they can only annotate 15% of the open reading frames from these wastewater phages. By using color-fold, predicting the structure first, and then using fold-seek, they could go up to 60%. So actually, you can now make sense of many of these open reading frames. Another study is by the Benfield group. They looked at these giant bacterial genes. There are these genes that are 80,000 amino acids long. So that's insane. That's much bigger than Titan, right? That's much bigger than what we have in humans. So the question is, why do bacteria have that, right? It seems very expensive. But so they wanted to understand, what does this huge thing do? So they took the 80,000 letters and broke them into pieces and tried to annotate these pieces. And you can see many of them can be annotated actually through classical methods. But whenever you see this donut here, these are regions where they have used color-fold and fold-seek in conjunction to actually annotate that and figure out what that region is. And you can easily use fold-seek by going to search.foldseek.com. You can just add a structure that you want to search. You can search through all of these different databases. And then once you search, it should hopefully take not too long. If it takes more than a minute, please contact us. And you can see all the databases that we search through. And you can see the 3-dimensional alignment as well as the amino acid alignment for each of the hits. Okay. Color-fold is actually, so we have a solution for metagenomics, right? So we can improve significantly in metagenomics. The problem is, when you deal with metagenomes, you have to deal with millions or hundreds of millions of proteins. AlphaFold is too slow for that. Here we have predicted a proteome of 1,700 proteins. It takes 4,200 hours for AlphaFold to predict that. With ColorFold, we are about 100 times faster. So it seems like, we're getting there. But that would be still really problematic to millions, right? So we need something that is faster to somehow really give the potential to the metagenomic community to annotate their difficult protein sets. And one solutions are language models. So you can see language models transformers come classically from the task of translation, right? Where you go between two languages. That's what the initial transformer paper actually described. You can have a translation where you go from amino acids to 3DI, 3DI being our structural sequence, right? So just translate between the language of amino acids to the language of structures. And this is where Burkhard Rost and Michael Heinzinger have helped to train that system. It's called PROST-T5. You can give it amino acids and it will print out, it will give you the predicted 3DI string. And now the question is, do we lose a lot? In our benchmarks, actually, you see that these predicted strings are nearly as good as the real structures. So there's not much loss that you have here. But the advantage is that this is actually substantially faster than using AlphaFold for predictions. So how much faster is that? It's about 300,000 times faster than AlphaFold 2. So now we are getting close to where we have to be for metagenomic scale. Still not here, still too slow, even though we are that much faster. But we can now do millions of proteins probably on a decent GPU set, right? So we are still working on making this even more fast. And I think there was also in a previous talk the mention about like smaller models. I think smaller models are really, really critical. Bonnie Berger, right? You said it. Yes. Yeah, exactly. We need smaller models. So. Yes, we have a talk. So yes, we need smaller models. These transformers are really big. They're billions of parameters, but you can actually train them much, much smaller and they would not lose much in performance. So that's what we have seen by trying to reduce the PROS-T5 transformer into a smaller system. It's still work in progress, so we have still something we work on. So for metagenomics, however, now, if you get faster in this prediction of the 3D strings, fold-seq becomes the bottleneck. Fold-seq is too slow. Like I have mentioned, it's very fast. We can do comparisons very, very fast, but somehow it is too slow for metagenomic scale. So here comes in work that we do together with NVIDIA, where we try to speed up MMSeq2, which is the backbone of fold-seq for comparisons, using GPUs. And I don't want to give you too much details, it's very technical. Initially, MMSeq was built with a sophisticated way of like using an index structure and like building something very, very smart to be fast. Now with GPUs, we do complete brute force. So we just do like compute every possibility and we do it on a GPU. And that's fast. In the past, you would never do that. That is really not a great idea. But GPUs became really good in these matrix operations, right? So they're optimized for like crunching through many, many, many cells at the same time. So, and you can write really efficient algorithms that somehow utilizes the GPU at the end. Yeah, I don't try to tell you too much about this. This is a speed comparison, but fold-seq is about 4 to 27 times faster on a single GPU than compared to 128-core CPU. It's like a huge CPU system. So a single GPU can be as fast as 128 cores. This is a very nerdy slide. In that slide, we searched 128 structures against the AlphaFold database. And this is real time, what you see here. So I think it takes approximately, I don't know, I didn't count, 22 seconds or so in comparison to 120 seconds it would take on 128 cores. So this is then fast enough probably for the metagenomic scale. So we're getting there, we're getting to that problem. One last thing I want to mention is we have extended fold-seq to multimers. So fold-seq in default works just on monomer structures, right? Like comparing one monomer at a time. But a lot of biology happens through interactions, and we somehow want to like organize interactions. We see more and more data is generated in protein-protein interactions and large-scale studies where they run AlphaFold multimer on like millions of proteins to figure out if they're interacting or not. So we need a tool that is actually able to utilize this data and organize this data. With fold-seq multimer, you can do this. I just want to show it you visually from a web server perspective. Here we search a CRISPR-Cas system for predicted with AlphaFold multimer from a metagenome. Like we took a gene neighborhood, predicted the interactions, and we searched it against the PDB, our structural database. And what you can see now is, this takes time because the thing is really big. You can see the chain overlap between this big machinery. This is like multiple chains that are searched in parallel through the database, and you can do it in seconds. So you can now really look at this like quaternary space and see what is different between your CRISPR system to another CRISPR system that we have seen before in our public databases. Okay, for the sake of time, I'm skipping the clustering of the AlphaFold database. But in conclusion, so I'm coming from this world where we use amino acid sequences to predict all of these things, right? All of these functional residues, protein interactions, and so on. I think we can now really move that forward by replacing it with structures or language models in the middle to really boost the prediction performance. With this, I want to acknowledge everybody that helped with this work, and yeah, thank you for listening.
We may have one question. Oh, yes, please.
How are you handling
so we currently, so how do we handle them? So we use the so we cluster superposition vectors of each monomer, and we treat them as monomers, but find a similar superposition based on the alignment. I can show it to you. I need a figure for showing you how to actually do it.
Considering them separately, you're not doing a linker between.
No, we yes.
Good. Okay.
Okay, if you have any other question, we have plenty of time for networking, so please use it for personal questions. Thank you, Martin.
Thank you.
Let me introduce our next speaker. Our next speaker is Professor Yunan Luo from the Georgia Institute of Technology. Professor Luo specializes in applying machine learning to computational biology with a focus on protein function prediction and drug discovery. Please welcome Professor Yunan Luo.
Great, thank you for the introduction and thank you for the invitation. It's my great pleasure to share some of our recent research in protein function adaptation and protein engineering. So we know that protein perform diverse function, and this function diversity is encoded by the arrangement of amino acids. But the natural protein we observe today is only a tiny portion of the entire space of all possible protein sequences. And if we consider the protein with function adaptation, it's even a smaller fraction. So one of the first questions we are interested in is to annotate the proteins without any function annotations. And another interesting question is about this, the remaining dark space. So we are interested in whether we can design a novel and a functional protein in this space. So it's basically an inverse problem of the function annotation problem. So given a desired function, we want to design a protein sequence and structure to achieve this function. So I will present some of our recent research, and some of them will be presented in Recon this year as well. So a major technique in our method is language model, but for protein sequences. So the idea is very similar to the natural language processing. So in natural language, we have model like ChatGPT, and this model is trained by masking some of the words or tokens in the sentence and train the model to reconstruct these marked words. And similarly, in protein sequences, we can train a similar protein language model. And so this language model basically is a transformer-based neural network architecture. So we can use the existing sequences and sample a sequence and mask some amino acids and train this model to predict a probability distribution. So basically telling us which amino acids are more likely to occur at this mask position. And to make a correct prediction, this model has to learn the grammar or semantics of protein language. And it has been found in many recent studies that using this model, we can improve the prediction performance or generation performance for many machine learning tasks related to protein biology. And this is still quite active research area from the 2019 paper by Tristan and Bonnie, and to a recent paper probably this year, the ESM3 paper, so quite active research area. And people have found that protein language model can implicitly capture sequence and structure features to predict like mutation effect at the sequence level, and also to predict contact, protein contact, and 3D structure at a structure level. And this talk, I will talk about some of our work, how to adapt protein language model for protein function. Okay, so the first task, function annotation. So we have several multiple annotation systems to annotate the functional protein. So gene ontology probably the most famous one. And for enzyme, we have something called enzyme combination number, or EC number, to annotate the function for every enzyme. So basically, those systems have been developed by creating the data using like wet lab experiments, and people are interested in using computational models to expand the coverage of the annotation systems. So the setup is quite straightforward. Given a protein sequence, we want to develop a computational model to make the function annotation prediction. And there have been many methods, including the classical sequence search methods, and also some of the deep learning methods. And I also want to highlight 2 examples. So the UniProt and PFAN databases, so widely used databases in protein biology, they both worked with Google to deploy a deep learning model to predict the function annotation of proteins. And those AI predicted annotations have been incorporated into their official data release. So this really demonstrates that deep learning has been very useful in real-world application of protein function prediction. So one of the first things we want to do is to inject function knowledge to protein language models to help the prediction of protein functions. Because the original protein language model is trained on sequence data, so it only captures the sequence similarity. But for function prediction tasks, we want to also encode the function similarity. So one thing we want to do is to inject the information from the function annotation scan into the original protein language model, so their representation space can reflect the function similarity among proteins. So, so we have developed a model called CLEAN for enzyme function annotation. The idea is based on contractive learning, so which is very similar to the complex work in bonus talk this morning for drug screening. So basically, we can sample from the function annotation scheme, which is a hierarchical structure. We can sample proteins with similar function. And we use the protein language model to encode these protein sequences into a representation space, but we enforce their representation to be close to each other. And at the same time, we can sample another protein sequence with a different function and encode in the same space, but this time we require their distance to be larger. So this is the idea of this method. And we found this works quite well for enzyme function annotation. And since its publication in Science 2 years ago, it still remains as one of the state-of-the-art methods for enzyme function annotation. And we also have some very interesting wet lab validation results. So our collaborator, they pick up a understudied enzyme. And we go to UniProt, and the database annotates this red function for this enzyme, and we run the existing tools like BLAST or some deep learning method. They all predict the red function for this enzyme. And when we apply our model, CLEAN, it predicts 3 different function labels for this enzyme. And in the wet lab, our collaborator actually observed this enzyme can actually perform these 3 different functions. Okay, so with this result, we are thinking about extending the machine learning based methods to other function annotation tasks. So including those gene ontology, protein family, and domain structure annotation. And one common problem, one common challenge we observe is a long tail distribution in those databases. So if we plot the class occurrence based on the number of proteins associated with different protein, we notice that there is only a few function classes associated with the majority of the proteins, which we call head class. And the other remaining function classes are sparsely allocated, and we call it tail classes. And actually, there is an initiative in the community, and which call for more research resources and focus to characterize the function of those head class, tail class proteins. So they, in a correspondence they publish in Nature Biotech and Nature Methods, they noted that there is about 95% of research papers focus on just 5,000 of well-studied human proteins, and there are many more proteins have not been annotated yet. And this is a rich get richer problem because new papers tend to target the proteins that have been already well-studied. So this is an imbalance issue in the existing database, and it also becomes a key challenge in machine learning modeling. So if we use enzyme function annotation as one example, I show the distribution. We have some head classes and tail classes. If we train a machine learning model on this imbalanced data set, we have some interesting observation. So we can get some good, reasonable performance, prediction performance for the head classes, but the performance drops significantly on tail classes. So this is somehow like a paradox. We want to use machine learning to expand the coverage, to reduce the annotation gap in existing database. But this gap itself becomes the key obstacle for machine learning to reduce the gap. So what's the reason? So we can understand the reason from the loss function of a machine learning model. So in the training of a machine learning model, we want to minimize the difference between the prediction and the ground truth, and we can sum over all the difference as a loss function to train the machine learning model. So we can decouple this overall loss function into two parts, one for the head classes and the other for the tail classes. So since we have many samples in the head classes, when training the model turned to overly focus on optimizing the prediction accuracy for those overrepresented head classes in the training data, and have very suboptimal performance for the tail classes. But what we want is to train a model that can allocate equal learning capacity to every classes in our training data. So how to achieve this? And we took inspiration from a paper from David Donahoe's group. So this is a machine learning theory paper, and they made a very intriguing observation in this paper. So they tried different deep learning-based classification models and train on different data sets, and they found a common property, which is that at the terminal phase of the training, the feature or the model representation of different samples from the same class tend to collapse to the center of this cluster. And the center in representation tend to separate with each other with equal angle. So this is a very special geometric arrangement of the features representation learned by this deep learning model. It's an equal angular arrangement. And very interesting, they observe this phenomenon for many high accuracy neural networks trained on balanced data across different tasks. So it's a general property. But on imbalanced data, this phenomenon called neural collapse does not show up. So we have a hypothesis. So since the authors of this paper, they made such observation on imbalanced data, high accuracy model often naturally exhibit neural collapse phenomenon, we hypothesize that on imbalanced data, if we can force the neural network to exhibit this neural collapse phenomenon, maybe we can have a high accuracy model. Okay, so from a very imbalanced parameter, very imbalanced representation space to a more, we developed a model called MSRAP based on this idea to induce neural collapse in the embedding space of a protein language model. So we use code the input sequence, but in the embedding space, we induce this neural collapse property in the representation space. And there are two important properties of neural collapse that we want to induce. The first one is called inter-class equal angularity, which encourages a model to learn representation for class centers to be maximally separated. And we have a loss function to encourage this property so that the class center can maximally separate with each other. And the second property is intra-class variability collapse, which encourages the embedding of the samples from the same class to collapse to the class center. So we have combined these two loss functions to train to reorganize the embedding space of a protein language model. And we found that this is very effective and improve the annotation accuracy across different protein function prediction tasks, especially for those tail classes. Okay, and we will present this work at Recon in a few days. Okay, so this is the first part. And the second part, the inverse problem. Okay, so now we are given a function and we want to design a protein sequence or structure to achieve this function. So how to do this? And And the protein fitness landscape is helpful in this case to conceptualize this process. So we can use a landscape like this, where the coordinate of every point in this space representing the protein sequence, and the animation of every point signifies the protein's ability to perform a certain function, or we call it a fitness, such as the binding affinity or expression. Okay, so how to find a functional protein in this landscape? This is a sequence search problem. And in biotechnology, there is a technique called protein engineering, which we can use to optimize protein function. There are two major steps in protein engineering. The first one is to identify some promising sequences as starting points, which has some initial activity. And this is a starting library design problem. And in the second problem, we can do fitness optimization. We can iteratively optimize the protein sequence such that it can reach to a desired functionality level. Okay, so and for the first step, it's quite challenging problem. First, we have a very huge search space. It's combinatorially large, and we cannot do exhaustive search. And also, this is a co-star problem. We may be interested in developing a new-to-nature function. We don't have any function labels to start with. And also, this landscape actually contains many hosts, meaning zero or extremely no fitness. So if we do random search, we may not identify functional proteins. So how to do this? So we want to use machine learning to guide this starting library design. And our goal is to design a library of protein sequences that are both functional and diverse. So let's first see how people are doing this using conventional approach. So in wet lab, there is a quite widely used approach called NNK library. So from a computational perspective, this is basically like a random search. As you can expect, many sequences would be non-functional in this library. But in our approach, we want to design a library with two objectives. So first, the library need to have a high expected fitness. And at the same time, we want to have a high diversity so that the library can contain some novel protein variants instead of collapsing to some single mod. And importantly, we want to balance between these two objectives. This is equivalent to balancing the exploration and exploitation in this design process. Okay, so for the first goal, how to quantify the fitness. Again, this is a co-star problem. We don't have any label data to train a model like a machine learning model. So our solution is to leverage a pre-trained protein language model. So as we talked about, in natural language model, the model can predict the probability of the word that can occur at a certain position. And similarly, in protein language model, we can train this model to predict the probability occurring at a certain position. And it has been found that this probability distribution correlates well with the experimentally measured fitness because it captures the evolutionary plausibility of an amino acid, so which mutations are more tolerable or beneficial at a certain position. And we use this idea, we use this protein language model to estimate the fitness for protein mutants. Okay, so this is the first part. And the second part, we want to achieve high diversity. So how do this? So we formulate this question as a Pareto optimization problem where we want to co-optimize both the estimated fitness and the diversity. The diversity is quantified using a radio level sequence entropy. And we design a gradient descent-based method to optimize this problem, which can be solved very efficiently. Okay, so let me show some results. So first, this is a visualization of the library designed by our model called Modify, and the conventional NNK approach, which is a random search approach, and also the FuncLib, which is a gradient search approach. And you can see our model prioritize the sequences that are functional and are at the same time very diverse. And we also experimentally validate this method in chemistry to design enzymes to catalyze chemical reactions in biocatalysis. And So we design a library, starting library using our method, and our collaborators validated those designs in their reaction system, and they found that our variants can deliver higher yield and selectivity in the reaction products compared to the conventional NNK approach. And they also noticed that the AI-designed variants are very far away from the human-designed sequences. So basically it shows the AI can give some novel designs for this problem. And we report these results in a Nature Communication paper, and we will also present the more results in a Recon highlight talk in a few days. Okay, so this is our solution for the first step, starting library design. And we have a second problem, so the fitness optimization. So once we have some initial sequences, how do we iteratively optimize the sequence to achieve a higher, a better protein function, protein property? So the key question here is to is like a hill climbing problem in this landscape. So at a certain point, so which direction should we go? So this is a quite hard problem, but for human, but nature knows how to do this through the long history of evolution. And actually, in a paper published by Maynard Smith in Nature quite a few years ago, he argues that if evolution is to occur, then functional proteins must be surrounded by other functional proteins. So this gives us some idea about how to identify functional proteins. We can do a local search. So starting from the initial sequence we have from the NAST method, the starting library, we can do a local search by introducing one or few mutations into this starting sequence and experimentally characterize their fitness, okay? And then we can identify a few very promising sequences, maybe at this position, and then we can repeat the same process to introduce a few more mutations and then repeat until we reach to a desired functionality level. Okay, so basically this is the exact idea of a method called direct evolution, which has been recognized by Nobel Prize in 2018 in chemistry. So this direct evolution is quite successful in many applications, but still it requires significant experimental effort. So we are interested in using machine learning to guide or to accelerate this direct evolution process. And one key question in this pipeline is to model the sequence to fitness relationship using a machine learning model. So basically, if we have the mutagenesis data characterizing the fitness for mutated sequences, we can train a supervised machine learning model. So if we have a such model for any given position in this landscape, any mutated sequence, we can run this model to predict its fitness. So this can be a surrogate of wet lab experiment, accelerating the whole pipeline. But this, again, a very challenging problem because we have limited data. So in one study of protein fitness characterization, scientists can often generate only tens or hundreds of variants. But modern deep learning model or machine learning model are very data hungry, often requires hundreds of thousands training samples to train an accurate machine learning model. So this requires us to develop a more data efficient machine learning model to learn this protein fitness landscape. And our idea is that instead of training a model from scratch, we can start with some pre-trained machine learning model with strong provider of sequence patterns in functional proteins. And again, we leverage a protein language model. So for a given position, given mask position, protein language model can predict a probability distribution. And if we compare this predictive probability distribution with the fitness data for various mutations at the same position, we may observe a good correlation between the model prediction and the experimental data. Okay, they correlate quite well. And this is not a single example for this protein. it has been observed that protein language model can give very good zero-shot fitness prediction across different protein, across different functions. Okay, so this basically tells us pre-trained protein language model are very good unsupervised predictor for fitness prediction. Okay, we have a very good unsupervised model. So it's very natural to reason that if we have some supervised data, we probably can make this model even better. So how to combine a pre-trained unsupervised model with some label data? So the first very natural idea is to fine-tune this pre-trained model by copying the weights in the pre-trained language model, and then train another regression layer on top of the pre-trained model. Okay, so we call it top layer fine-tuning, and we try this idea, and the performance was improved a little bit. So a good thing of this approach is that it's very robust because we only need to train a small number of parameters in the top layer. But the disadvantage is that the model capacity is quite limited because we are not able to fine-tune any parameters in the frozen protein language model. So how about fine-tuning the entire protein language model? This is another natural option, and we also try this. And interesting, the performance is very, very, very poor. So even worse than the unsupervised prediction. So this suggests a very significant overfitting issue. So what's the reason? We realize that the key reason of this overfitting issue is a discrepancy between the pre-training stage and the fine-tuning stage. So in the pre-training stage, we have a maximal likelihood objective. So we want to reconstruct the true amino acid at a position using a maximal likelihood approach. But in the fine-tuning stage, we use a regression training objective. So the switch of the objective function makes the model force the model to reconfigure its parameters learned from the pre-training stage, so which destroys the sequence patterns or prior knowledge it has learned in the pre-training stage. And this is known as catastrophic forgetting in machine learning. And also we have a small data problem here. This makes the problem, the issue even worse. So we provide a new method to address this challenge. And our idea is like this. So since the predict probability distribution correlates with the fitness data reasonably well, we can preserve this output space in the fine-tuning stage, but calibrate it to better correlate with the fitness data labels. So let me show one example. So we have a pre-trained protein language model and two label data. So we have a sequence with a red mutation, which has a higher fitness, and another sequence with a blue mutation, which has a lower fitness. So in the output space, output layer, we increase the probability for the red mutation, for the red amino acid, and decrease the probability for the blue amino acid. So in this way, we make the output of the protein language model to be consistent with the ranking of the ground truth fitness data. So this is also a contractual learning problem. And very interesting, we found there is a classical statistical model called a Bradley-Terry model, which can capture our idea and use it as objective to fine-tune the protein language model. And we trained this idea, and we found that this model can realize the full potential of the protein language model, and at the same time avoid overfitting. And we observed a very significant improvement across over 30 protein data set, and even using less than 100 mutants as training data, we can predict the protein fitness landscape quite accurate. So this is a method so-called confit that we can use to adapt the general pre-trained protein language model, which captures the general evolution landscape, into function-specific or protein-specific fitness landscape. So even using a small amount of data. Okay, so in Recon, we will present a related work where we integrate multimodality pre-trained protein language model for protein fitness prediction. And if you attend Recon, please attend this session. Okay, so to conclude, we introduced a few methods for protein function annotation and protein engineering. So the AI methods in other applications like computer vision and natural language processing are very successful, but oftentimes if we directly apply those methods into biological problems, they do not work quite well because there are unique challenges in biological problems. So the annotation bias, small data problem, and the color state challenge are some of the examples, some of the unique challenges we have in biological questions. And we need to tailor those AI methods into those biological problems. And in this talk, we show that protein language models could be effective solutions for those unique challenges. And if we can reprogram or customize those pre-trained protein language models, they could be very good solutions and improve the prediction performance or design performance quite a lot. Okay, with that, I'd like to thank all my students and our funding agency, and I'm happy to answer questions. Thank you very much.
We may have one question. If not, thank you, Professor. Oh, okay.
So I was wondering if you could look at the directed evolution, like a former speaker was talking about causality in some kind of a way.
Yeah, I think that could be a quite challenging problem. So because the search space is quite large, so there are multiple evolutionary paths from a starting sequence to a final functional sequence. So if we can constrain this search space to a manageable space, maybe we can explain or identify the causality path for those evolutionary history. But still, I think it's quite a challenging problem due to the large, huge search space. Yeah, but thank you for your question.
Okay, thank you, Professor Luo.
Thank you.
Our next speaker is Professor Chao Seok from Seoul National University. Professor Seok is renowned for her work in computational chemistry, particularly in protein structure prediction and drug design. She leads the development of popular protein modeling programs, which are widely used in bioinformatics research. Please join me in welcoming Professor Chao Seok.
Thank you for inviting me to this wonderful conference. Actually, I misread the program and I thought that my time is 10 minutes, but then now I learned it's 30 minutes and I quickly amplified the number of slides. And so some of the later slides have some Koreans, but please excuse me for that. I have two affiliations, Department of Chemistry, Seoul National University, and I'm also CEO of Galax Inc. And today I'm speaking as a CEO of Galax. And we are very proud that we have some successful cases in de novo antibody design. And because we are a company, we cannot reveal the details of our algorithm, but it's very exciting results. So I want to show you them. And we have two recent bioRxiv papers. And the first one published last year, we submitted last year, is CDR redesign. So we only redesigned the let me see. Can you see? Sorry. Well, so we redesigned this brown part. Can you see on the lower left corner? Only the CDR loops of the antibody. Given that we have the complex structure of antibody and antigen. So that's very strong information, but because we were not pretty confident about our ability at the time, we started with the CDR redesign at as the first work. And then next, after confirming that we are pretty successful with CDR redesign, we attacked the task of de novo antibody design, which, let's see why this is moving. So here, we don't use any prior knowledge of known antibody structures or sequences. We only use the antigen structure. The antigen structure can be either, can be either experimental structure or the modeled predicted structure. Oh yeah, so sorry. So it is supposed to be a movie that the antibody structure appears on top of the antigen structure, but it's not showing here. So we specify the epitope where we want to bind the antibody, and then we design the antibody in de novo design. So after we submitted this to bioRxiv, we had a lot of both positive and negative reactions. Ian Wilkinson is one of the influencers in LinkedIn. I'm not doing. I don't have this any activity. I'm not active in LinkedIn, but I just heard that that person posted this message about our paper, and then more than 100 replies were, there were more than 100 replies, and some of them were very positive, and some of them did not believe. So we want to generate more experimental data to convince them. But anyways, there are more than 50,000 impressions on this post. So I think we are successful in attracting attention, and we'll see how it can lead to a real drug discovery. So our company is based on Galaxy protein structure prediction program. I have been developing this in Seoul National University for more than 10 years, and if I begin my career, count my career from my postdoc years in UC San Francisco, my protein structure prediction work has 25 years of research. And then based on this experience, we founded Galax, which means galaxy plus looks light. So our co-founders are all from our lab. Okay, and now we have both dry lab and well lab. Okay. So this is about this is not business conference, so maybe I should not talk too much in detail. But our business model is to have some flexible partnerships with different types of pharmaceutical companies by using Galax design, protein design platform. So in order to do that, we have to first translate medical unmet needs to molecular unmet needs. That sets us with a goal for the molecular property we want to achieve. So that's called the TPP, target product profile. And then we design proteins based on our generative model, which is based on also protein structure foundation model. We have some antibody-specific generative model fine-tuned from our foundation model and so on. So we design those molecules trying to satisfy the target property, and then we do some screening in the lab and produce proteins in lab scale and do some experimental validations. Okay, so the first part is about antibody CDR loop redesign, and the second topic is about de novo design. And the first work was done with Kellogg's design versions 1 and 2. So this part, our main strategy was to improve the protein structure prediction accuracy, is in particular in the loop structure prediction accuracy. So the performance of Kellogg's version 1 and 2 in 83, which is the most complicated, flexible, variable CDR loop. Out of 6 loops, 83 loop is the most complicated one. And for that, we had the best structure prediction accuracy. So the lower RMSD is better. And our set is newly generated. The GFD set, it means Kellogg's antibody data sets, which is composed of more recently discovered structures. So it's more difficult, and the numbers you see is rather high for the well-known antibody structure prediction models. So that's why they are higher, because we have more difficult and more recent data set, which does not overlap with the training set of those methods. And then on the right, I show you the in silico design success rate. It is very hard to assess, to evaluate design methods because without doing any experiment. So how do we evaluate in silico design? There's no perfect answer. So we tested the design performance in 2 measures. One is structure recovery, and structure recovery is the percentage of the designed cases where the designed protein structure has similar structure to the known antibody antigen complex structure. So, so you see the numbers, and Kellogg's design has higher number than other well-known methods. Here, the RF diffusion is not the antibody-specific one. At that time, we did not have the RF antibody available. So anyways, the second measure was GPass, which just measure the quality of the designed sequences in terms of the predicted structure. So we confirmed that our method is improving in silico, also in the scoring, discriminating binder and non-binder. Then we move on to the in vitro validation, where we tested, we produced those designed antibodies in IgG form or a single chain SC FV form, and then screen for binding. And for similar task, this company called AIxBIO in bioRxiv 2023 paper, they obtained a success rate of they report their success rate is 1.8% in redesigning the antibody loops targeting HER2. So the antibody, the name of the antibody is trastuzumab, and we redesigned the same antibodies, and our success rate of finding binders was higher, much higher. Okay, and then our version 2, for different target, our version 2 has higher success rate than version 1. So that was very exciting. And also we were very excited to see that our redesign method could find an antibody that can discriminate single amino acid mutation in EGFR. Okay, so some details. I'm showing that our sequences are novel and things like that. Okay, so so let's move on to the de novo antibody design. Before us, there were 2 reports saying that they succeeded in de novo antibody design, and the 2 are Nebula Bio, a US-based company, and David Baker Lab. And Novotel Bio reported the antibody designed against SARS-CoV-2 RBD with binding affinity, KD, the dissociation constant of 30 nanomolar. To be a commercial antibody, therapeutic antibody, the dissociation constant should be less than typically 1 nanomole, and so the dissociation constant should be like picomolar affinity. So this is rather high, but it's very exciting that experimentally validated to novel antibody design. And then David Baker reported that they succeeded to generate antibodies for 2 targets with 70 nanomolar and 400 nanomolar affinities, which are rather weak. And sorry, this is supposed to be Galaxy Design version 3. Okay, so our design is just a typical de novo antibody design. We first assign the epitope residues, usually 2 amino acids to 5 amino acids, to modulate the function of the target protein. And then we generate candidates and then select it based on our own scoring. And we succeeded in, we experimentally validated that we found antibodies for 6 therapeutic targets shown over there. And we did this by designing library and then performing yeast display experiment in the wet lab. So the antibody has some complications because it is composed of heavy chain and light chain, and finding the right pairing is not easy. So basically, we designed 100 heavy chains per 100 light chains, but the total library size is 10 to the 6 somehow, because we have 10,000 heavy chains times 100 light chains, so it's 10 to the 6. So anyways, for some of the cases, we validated individual antibodies by producing them and then purifying them and so on. But we are still working on other targets. So we have some more data for PD-L1 case and less data for other cases. This is in silico performance comparison for our Galaxy version 3. So the GPAS is for de novo antibody design, and you see that our success rate, in silico success rate, is continuously increasing. And in other tasks on the right, we see the orientation recovery. So this is not like exactly this does not exactly correspond to the real performance, but it's just an indication of the performance, the structure recovery. We have higher rates than other well-known methods, such as RF antibody. Okay, so this is our scoring performance in terms of AUROC, and we have higher success rate in version 3. So this is the first example showing that we have very specific antibody that has like commercial the properties comparable to commercial antibody called atezolizumab. So you see that the titer is very high, and our KD is the best one, has 9 picomole. So it's extremely strong affinity. And in the competition assay, we confirmed that our designed antibody compared with atezolizumab, meaning that it targets the exact epitope we were intending. And we are also trying to solve cryo-EM structures. So it's underway, and the thermal stability is very high. Monomericity is high. It does not react with many other known targets. Polyreactivity is none. And we also performed 2 types of cell binding assay, and it was very good, comparable to the commercial antibody. So this was the PD-L1 case, and the detailed data are shown here. Yeah, I'm skipping this part. And then, so another target, this is also, sorry, the many of them are in Korean. So this is also a proof of concept target, EGFR S468R. And here we could confirm that our designed antibody binds the wild type EGFR and does not bind bind the mutant and does not find the wild type antibody. So that's the last one. And so this one, actually this mutant is very easy to discriminate because the mutation is from the small polar amino acid serine to large positively charged arginine. So our designed antibody could easily discriminate the mutant and wild type. And then another interesting example is design on the previously unexplored epitope. So this new epitope is very important because it can cure, it can overcome the difficulties people had in clinical failures. Because the previous antibody therapeutics failed in clinical tests because the antibody bind Frizzle7, and also the antibody interacted with other subtypes of Frizzle family. So you know that the Frizzle7 is cancer target. But if the antibody interacts with like Frizzle1, then you can have like problem in bone fracture or something like that. So sorry, this all is in Korean. But the problem is this epitope has very similar structure in different subtypes, Frizzle subtypes. But we could so the main difference is in only in two amino acids, and our design was able to discriminate Frizzle7 and other subtypes, as you can see on the right. The previously known antibody could not discriminate different subtypes. So that was a very interesting one. And then the last interesting one is AIK7 target. This is related to metabolic disease. So this one, if we have a successful antibody for targeting this protein, we can have maybe a better drug for weight losing or something. Okay, so here we used the predicted structure for ALK7 because we did not have any experimental structure available, and we specified the epitope at the interface of activin and ALK7 to get required function modulation. And we were successful in finding antibody that binds only ALK7 and not other proteins. And then now we are also testing these antibodies, whether they really block this function and so on. Okay, so far, our company succeeded in CDR redesign and then de novo design in several cases. So we are expecting to go to market pretty soon. Okay, thank you for your attention.
Thank you. And any question? Okay, then Thank you, Professor Seok. Thank you to all our speakers. Now, we completed all the sessions before the banquet today. At this point, I would like to invite all participants for a group photo shot at the front. Please join at the front. Yeah, another Korean style. Thank you, everyone. And before the banquet, I would like to invite Professor In-Seok Lee from Yonsei University, who is the ex-president of KSBI, for closing remarks today.